finishing the material that we are supposed to cover for the test. And today in the lab, I'll answer any questions that you have. Then you would have it available for you to do the test online. Now, so we started from the regular introduction to biology, and we went right up to talking about the cardiovascular system. That is the first system that we talked about. And when we talked about the cardiovascular system, we talked about blood, talked about some blood cells, some of them being white blood cells. So it just kind of fits that the next system that we should look at is the lymphatic system, which is also incorporated to what we call the immune system, that also deals with some white blood cells. So we will start today talking about the lymphatic system, and you should have the slides available. You should have access to them now, but I will share my screen for us to start. But before we do so, does anyone have any question once more? Can you see my screen?
Yes.
Okay, I'm going to get us going. All right, I'm going to share my screen. So I said by the time that we are done, we're going to talk about you know what the lymphatic system is and what it is made out of and what its function is. And as I indicated, one of the things that we'll be talking about that is incorporated in the lymphatic system is our immune system, how our body defends itself from pathogens, things that want to get into it and make it get sick, or things that originate from within to make this bodies sick. And to do that, we will talk about microbes, which are things that you can see already under the microscope, and how they relate to humans. We know that we'll talk about some beneficial uses of microbes, but of interest, we'll talk more about the microbes that make us sick, which we'll refer to them as pathogens. And when we are looking at how the body defends itself, we're going to see that it is almost like it has different levels of defense. You will hear about talking about the first, the second, and the third line of defense. And then we'll talk about in this third line of defense, we'll talk about the cells, the white blood cells and antibodies that are involved in it, what they are, and how they do their job. And then we'll talk about B cells, which is one of the kind of white blood cells. And then we'll talk about immunity. When we had vaccines like against COVID, we said, oh, we're going to, the one herd immunity, many people should take the vaccine. So what is immunity? And then we'll say some is active immunity, some of it is passive immunity. We will be able to know, you should know how to describe the difference. And then we'll talk about allergic reactions. I don't know whether some of you have allergies, what is involved in the process. And then we'll talk about some aspects about, when they do like transplants. We're talking about the blood types where I said I cannot give blood to this person, or this person can take blood from this person without any problem. Sometimes you might also, there's situations where they have to either give tissues or organs to other people. But some people would ask, they say we don't have the right match for that individual. So if they don't give things that, or tissues or organs that are the right matches, there's tend to be some rejection. Their body fights them. So talk about tissue rejection. And then finally, we'll just kind of summarize and talk about some problems or some disorders that are related to the immune system. So I think, let me see whether I should hit this video. We take a quick look at it. But I think even before we do that, let's see here whether we can see this. Do you see my screen and do you hear sound? Do you hear no sound? Okay, that's interesting. All right, let's take two. Go back here. Go to the any sound?
No.
All right, let's not worry about that. You'll have access to the video later on, but will play it later on. So I'm going to kill this. Go back here. So we talk about microbes. I said, what are microbes? From the way the word sounds, is things that are too small for us to see with the naked eye and would require a microscope to be able to see them. And these would include bacteria, viruses, and then some proteinaceous material they call prions, and some fungi. We know fungi include mushrooms, which we can see, but we are referring to things like yeast, and then we are referring to things like some like other kind of small fungi that is too small for us to be able to see. And then we know things like amoebas, which we were some of them when we were looking at things under the microscope the last time from pond water. You see some organisms were moving in there, or some were very small. Those are again, some of those things are things that we could generally of interest refer to them as microbes. And putting things in terms of scale, for relative scale, if you look at the size, this is our prototype of what we say the eukaryotic cell looks like. And if you can aim this in terms of size, then you juxtapose, put them side by side with that of a virus and a bacteria. Bacterium is one. So you could see that generally they, compared to viruses and bacteria, our cells look huge. They said one virus, an example, adenovirus, is 0.009 micrometers, which is 19 nanometers. Then a bacterium is around 2 micron. And then our red blood cells is 10 micron. So our red blood cells is much, much bigger than E. coli, for example. And then paramecium, which some of the things, if you had, normally we would have seen some of them in the pond water. It's about 200 micrometers. Then forget in terms of size, look at the mushroom, much, much gigantic. A person compared to just huge. So we know that we interact with because in our environment, we have bacteria. On our skin, we have bacteria. Inside us, in our stomach, we have bacteria and even viruses. We know that some of the bacteria, they serve a purpose for us. Some of it good, some of it not so good. And I put here, I said, microbes are very abundant in the environment and in our bodies. We use some microbes to make food, and we even use some to make drugs. And we use some microbes are also involved in decomposition of dead material to recycle nutrients. Let me just throw this back to you. What are some of how do people use microbes for food, for example? How do we use microbes for food as in our food processes or as food? Any ideas? Or do we use, or is this sentence wrong that we use microbes to make food, some food material, food stuff for us? Antibiotics, yeah, and fungus for antibiotics, that is for drugs, yeah, that's great. Like penicillin, they make it from fungi. Which one? We use molds, yeah,
You say yeast like in bread?
absolutely. Yeast, we use yeast to make bread, and because yeast, when it undergoes anaerobic respiration and produces carbon dioxide, so it makes the bread to rise. And then when it rises now, it expands, and then the dough that you knead, and then it makes, and then you can now make bread with it. Any other ideas in terms of food or drinks? Wine. When they make wine, they make beer, they use the yeast again to do that. Because in that process of fermentation, one of the byproducts is alcohol. So in making bread, we take advantage of that product of that process of fermentation, carbon dioxide, to make the yeast to make carbon dioxide to make the dough to rise. In making wine and in making beer, the fermentation process, the benefit of that of using yeast in that process is they're looking more after the alcohol. So I'll ask you a question that if you look at the fermentation process, meaning yeast would ferment sugar in the absence of oxygen to produce carbon dioxide and alcohol. What happens to the alcohol that in the dough when they make bread with it? What happens to it? Why is it that when you eat bread, you don't get drunk? If the process that it was being made had yeast in it, and then the yeast was able to make the dough to rise, and because of the carbon dioxide, and in the process it also produced alcohol. Why is it that when we eat bread we don't get drunk? Any thoughts?
Does it like evaporate when it's getting cooked?
Exactly, because alcohol tends to evaporate, and when you put it in the oven, it evaporates in the baking process, and then you don't get drunk as a result of that. So, and then in terms of decomposing, when organic material, when animals, plants, they die, they have to break the tissues are broken down by bacteria back to the various elements, and carbon and all of that, and nitrogen and everything, and it go back to the basic elements. So if you look at it, if imagine that there was a world where things die and they don't decompose, it will be horrible. But now, when farmers when they plant their stuff after harvest, they will tilt and mix everything up. It decomposes, it becomes a manure, and then the plants use that again to plant new plant seeds, and then the use of the manure, and the process cannot go. So they serve a very crucial role in our environment, um, related to food, related to drug making, decomposing. We now even know that there are some microbes that would eat plastics, and because we're having environmental problems of plastics, and we say like in the oceans we have plastics, plastic waste, plastic bags, and we don't know what it takes a long time because they didn't have a way of recycling them. But now they've found some microbes that are able to feed off this material. Oil spills. When we have oil spills, you cannot you know that you can take detergent like palm olive or others to be able to break down the oil. But imagine that you have an oil spill in the ocean. You don't want to take like palm olive soap to try to break down the oil. But now there are some microbes that are there that can eat all those 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 hydrocarbons, the oil, without actually causing more havoc in the environment. So if you look at all of these beneficial aspects, however, there are some microbes that cause diseases in humans, even in other organisms. And the general way we refer to those as pathogens. Pathogens. Give me an example of some microbes that would make us sick. What causes sore throat?
A virus?
Uh, Not quite. It's a bacteria. Some people say have a strep throat. They're referring to the bacteria that causes. I put here some human pathogens. So we have viruses, we have bacteria, we have fungi, we have protozoa, we have some parasites, we have even some proteins. So, like for a virus, let's take the bacteria that I mentioned that first. Bacteria, as we talked about it when we were talking about types of cells and things like that, bacteria are prokaryotes. They are usually single cells, and they live on our faces, on our entire bodies. We have them inside our nostrils, we have them inside our stomach. So there are some that do not cause us any problems. However, there are others that would make us sick. We have one, tuberculosis, the one that makes people cough. We have anthrax, we have staphylococcus, we have E. coli. And if you hear E. coli, you say, oh, E. coli have different strands of it. So strand is like saying a different um, variation, mutation of it. Some of them will not cause problems for us. Some of them do. Salmonella. And then you hear for viruses. Viruses are, yes, they are also prokaryotes, similar to bacteria. But if you look at viruses, they are not considered to be actually living. But the interesting thing about them is that they carry out some of the other things that living things do. For example, they would replicate, but they need a cell. They themselves don't have the tools to be able to replicate. When they get into a cell of a human being, or of a bacterium, or of a plant, they use the cell's machinery to be able to reproduce. So viruses are considered acellular, as they can only reproduce in a living cell. So you have viruses that cause hepatitis, herpes, warts, chickenpox, smallpox, bird flu, the one we're talking about, monkey virus, coronavirus, Ebola virus. So some of what we have, like the colds that we have, are thanks to the viruses. Cold sores. Meanwhile, fungi, as we're talking about, you have like yeast infection, ringworms, protozoa. We have some that would cause malaria. They actually, and then we have we have, and malaria is like a fever. It makes people have like almost like have a fever, but they're caused by a protozoa. And then we have parasites like tapeworm. We usually don't have those in around now, but in third world countries where you could have in the material that is infected with the thing. A tape. How many people know what have heard about a tapeworm before? Anybody? A tapeworm.
Are those like worms in your stomach that are parasites basically and they feed off what you eat?
Yes, it's a kind of worm that gets into your stomach. Most of the common worms that we have in our stomach, that when you are infected with them, they give you medication, you go and poop them. It's almost like you are pooping spaghetti strands. They are like almost like that. And this is like, but a tapeworm is another kind of worm that is much bigger. The way how when you eat meat or food that is infected with it, it would go into your stomach and in the wall of your small intestine, and then it would burrow into the wall of it, and then almost like have its mouth open. So when you are eating food, the food it's absorbing instead of how you are after digestion, your food is absorbed. This one will be eating it and then getting bigger. And then sometimes you will see some kids who have tapeworms, they would have, they will be skinny, they will eat, but they'll be skinny, but their tummy will be getting bigger. And then, so all of this are things that we know that divide and either protozoa or either bacteria and viruses. But guess what? There are some other proteins that can also cause us to get sick. Proteins we know are made up of subunits called amino acids. If you have ever heard about mad cow disease, they usually, when they were trying to find out, okay, what is causing mad cow disease, for example, is it the test for bacteria, they didn't find it. The test for viruses, they didn't find it. The test for fungi, they didn't find it. Then they finally saw that there were some proteins that normally exist where we have our nervous tissue, like in our brain, our spinal cord, and the nerves. But if you eat meat or food that is contaminated with a form of this protein, it can the messed up form of the protein can go and convert the existing good proteins that you have in your nervous tissue to transform. And when they transform, they kind of gum up together and then make the nerve cells not to transmit impulses properly. Because one of the things that you notice with mad cow disease is that the animals, they kind of lose balance and all of that kind of coordination, things that their nervous system should have been helping to make them resolve. So I put here examples of some disease caused by pathogens. We talked about measles. We don't normally have measles now because we have when you are born, when you are born, you have this regimen of, oh, take this set of vaccines at this time. Just take this set of vaccines at this time. One of the vaccines that they make kids take is the one against measles. When you have a kid has measles, you have all this almost like blisters over all their entire body. It's caused by a virus. And then we have, first of all, since we just kind of talked about vaccines, we'll talk about that a little bit later. But we have bacteria. You would hear, you have your tonsils here. One is fine. But you see this one here that looks swollen. This one looks swollen, and this white stuff on it is pus. Pus is caused when our immune system cells are fighting the bacteria, and then they will die, and then they form this pus like this. And then, so this is like pus containing the bacteria, that streptococcus. Then athlete's foot. Again, if you play sports in the rain and then you have your shoes, you don't usually dry them, you just wear them back. Fungi like this will tend to grow in wet, warm, damp areas but it can cause issues for us. But these are just examples of some pathogens cause diseases. Just as a refresh, here, this is bacteria, a prototype of a bacteria. It looks almost like a corn dog. Bacteria have different shapes. Let me just put this up first before I go back to that. They have different shapes. Some of them can be round. Some of them can be round and by themselves. Some of them can be round and just kind of stuck together like in pairs. And then some of them can be round and then stuck together like in 4 groups or something like that. And then some of them could be like this corn dog shape. Some of them with conducts with a little bit of, these extensions like wiggling stuff like we call flagella on them. And some of them are not really like straight conducts, but they're like, twisted. They call them spiralis. So like the ones that are round, they'll call them coccus for one. And then there are 2 here, they say diplococcus. And then, so those that are around, they usually use the word coccus, and if there are many of them, they cocci for many. And then you hear this one like tetrads, streptococci, because streptococcus is referring to one, streptococci we are referring to many of them. And then the ones that are rod-shaped, you call them bacillus. And then that's like one bacillus. And then many of them you have bacilli. And then you have salmonella, that's an example of that. Clostridium is one of them. And then we'll say they have flagellates, they have flagella, these extensions on them. Then the ones that are kind of wiggly, we'll call them spiralis or spirala, spirochetes, Treponema pallidum. Now, we're going back to our bacteria here. We say bacteria are prokaryotes that are single-celled. Usually, they're made up of, they have a cell wall, which we do not have. But they also have a plasma membrane, which we share. And then some of them would have a capsule. And depending on the kind, they might have this, some of them might have this flagellum and have cilia on them. One of the things that, in terms of their nucleic acid, yes, they have a DNA, but unlike us, that in our cells that are prokaryotic, their DNA is not surrounded by a membrane. It's just like inside. Just imagine that you have like a bag, a tubular plastic bag, for example, and then you take some threads and then you shove them inside. So the bacterial chromosome is just has the DNA, is just kind of inside like that. But if you're going to compare that to our cells, our chromosomes, they are packaged surrounded by by this membrane to make in a nucleus. But of importance that I would like you to keep in mind is that besides the bacterial DNA, when I say bacterial DNA and turn around and say chromosome, don't get confused. Chromosome is, you're talking about when the DNA has attached to these proteins and then it's held together. So because the DNA is like a long thread, right? But now the thread is not just by itself. To make it fit properly, it wraps around some proteins called histones so that they can all fit better. So anyway, you sometimes hear our DNA or our chromosomes, it's just like the same thing, referring to the same thing. But now there is a separate DNA that bacteria have that are like this, they're circular and they're called plasmids. Some of the genes that bacteria have in the plasmids produce proteins that can make us sick. The ability of the bacteria to fight like antibiotics or something like that, they have genes on here that make them to fight off when we try to kill them. But they are also, as we're going to see, they're very important because these bacteria, you can use these plasmids to put genes into them and then transfer these genes, transfer the plasmid into other bacteria or into other cells. So we can use it almost like a transport of genes. We'll talk about that when we talk about biotech. And these bacteria, how do bacteria divide? They divide by a process called binary fission. So where you have one bacteria, when it wants to divide, it grows bigger. After it has divided its DNA, and then when the bacteria wants to divide, it gets bigger and then splits up into two. And then the two would split up like into four. Each one of them would do the same thing and then split up into four. The four would then split up into 8 to 16, 16 to 32, 32 to 64. And some of them do this, some of them can even do this like every 20 minutes. So you can imagine you can have one bacterium, and if you calculate that it is dividing every 20 minutes, that kind of growth where it divides, they keep dividing in this exponential way, makes bacteria to be able to populate an area very quickly. So they reproduce by binary fission. And then in terms of shape, they have various shapes, some are round, called coccus, rod-shaped, bacillus, spiral, spiralis. And what we use to kill bacteria are antibiotics. Example, penicillin. This one obtained from a fungus, penicillium. Now, when we talk about antibiotics as a way to kill bacteria, it is also, I think, necessary that we talk today about, mention the fact that there's been a big push to say, oh, we have some antibiotics that are no longer working. They say, oh, this bacteria type is now resistant to antibiotics. So what happens? What causes antibiotic resistance? So this is a picture that kind of shows how you can get some antibiotics that used to kill some kind of bacteria, and then out of a sudden it doesn't work anymore. Okay. So I said antibiotic use does not cause humans to become resistant to the drugs. So it is not us who become resistant to the antibiotic. However, it is the pathogen, the bacteria that become resistant. And how does this resistance happen? Imagine, we have not done this yet in the class, where you grow bacteria on a plate. When you grow bacteria on a plate, so after a while, when you take the plate, let's say we're going to do an experiment where we will take wipe our faces and then rub it on a round dish on something that like they call it agar, which is like how you make jello. Let's say you pour jello in a plate and then let it to harden up. The surface or the thing on which bacteria can grow in is something like that. It has nutrients that the bacteria need. So if you wipe your face, as in the experiment we're going to do, and then you rub it on the plate, after a while, it is going to create, you come to look at it, it will have like some spots. It's almost like you took some milk and then just kind of put droplets on it. And then all of those droplets you see are groups of bacteria. Now, when this plate here is representing bacteria, they are normally supposed to be identical, but in any bacteria population, there are some that might be just slightly different. What can make some of the bacteria different? Either some environmental chemicals can make some of them slight mutations in them, but generally they don't have too much variety in mutation. They don't have too much variety because the manner in which they divide is pretty simple. That process of binary fission, we'll talk about cell division a little bit more later, is very straightforward in the sense that the DNA will divide compared to the way our own cells divide. Our cells divide in a more complicated manner that introduces a lot of variation. But now let's focus now on terms of how the bacteria would become resistant. Imagine that all of these bacteria here, we have identified an antibiotic that can kill them. But the ones that are black would be killed easily by that antibiotic. And then these two that look a little bit orangey or brownish looking here are slightly, because of a small variation they have, they will not die quickly from the antibiotic. So this is a slightly heterogeneous population with these two variants. Now you apply the antibiotic to this. After you expose the antibiotics to this, guess what? These ones that are colored black here, the antibiotic will start killing them easily. They will start killing them easily, quickly. And then now, as they're killing them, these ones here that are not dying quickly, they will start dividing because in this condition where everybody is present, the bacteria, they are all trying to get food and get places to grow on. So they are limited. These ones are limited in terms of how they can grow because they don't have enough food. But guess what? If you kill most of these black ones here, the brown ones start saying, oh, there's excess food for us. They can divide too and grow more. So now, if you continue or you stop taking your antibiotics, say at this point, these ones would grow and divide more. So imagine that I said in a given population of pathogens, there are some organisms that are naturally resistant to the drug. They have acquired some resistance through mutations, which are changes in their DNA or interactions with other organisms. The drug kills susceptible, means that the ones are weak ones, but leaves the naturally resistant ones to multiply because they have more resources, food, space to be able to grow. The new population is then resistant to the drug. We are having issues now with tuberculosis, malaria, gonorrhea, staphylococcus. All these are groups that have become slightly resistant. But to illustrate the mechanism, we go to the second slide here. So again, imagine this bunch of bacteria here with one of these ones that is slightly messed up in terms of it's slightly different. When you challenge it with antibiotics, it kills all these other ones besides the one that has this mutation. When you stop taking the antibiotics, it allows this population to grow. But if you keep taking the antibiotics that a doctor has given you, for example, even though this one is trying to divide, but now the antibiotic has enough, there's more of it now to challenge this one, usually they will die. So if you say, for example, your doctor says, well, take this antibiotics and take it for 7 days. You say, okay, thank you, doctor. You go, because you had a strep throat. Day one, it wipes off most of these ones. Day two, 90% are gone. Day three, 99% are gone, but these ones still remain. And then you go to school and you meet your friends, say, how's your cold? Say, my cold is getting better now. And, I don't have the strep throat anymore. And then your friend says, well, listen, my throat is beginning to feel sore. He says, no, I'm well now, but I had some leftover antibiotics that my doctor gave it to me. And then at this step, because you are feeling your symptoms are gone, because only a couple of these ones or just one is remaining, you take the antibiotics and give them to your friend. Guess what? If these ones stay and it doesn't eventually die, it could create a new population that would have the more resistant ones, such that if you get sick, you don't have a mix of this one and this one anymore. You have more of just this type. And when more of these types that were, because of the had this mutation, if you were taking the same antibiotic, it's not going to work. So part of it is us having mutated bacteria in a population and not using antibiotics properly to be able to make sure that all the susceptible ones and the ones that are slightly difficult or not so susceptible, you don't use them, expose them in the first time around to make sure that you wipe out all of them. This is the one way that we have control of dealing with making sure that to be able to stem this from happening, we take antibiotics as prescribed all through, as the doctor said we should take them. The second way in which bacteria can become resistant to certain drugs are, if you look, let's say remember all some of these round things here are like the plasmids that I was telling you. Sometimes bacteria that are resistant to antibiotics don't cause us problems. Like when we take antibiotics, we swallow them. The bacteria in our stomach, they're just fine. Some of them are just fine because they got used to it. They have resistance to it, and they stay there. They're harmless to us. Or even on the surface of our skin, when you apply antibiotics on it, some bacteria that normally stay on the surface of our skin is okay. It doesn't bother. However, always ever so often, even on the surface of our skin, there might be a couple spotted harmful bacteria. But because it can be one or a few, their population is not enough to be able to produce proteins or products that will make us sick. But when harmless bacteria stick around with harmful ones, imagine this one is harmless and resistant to an antibiotic. This one is harmful but not resistant. If these stick around, sometimes the bacteria, they do a process called conjugation, where one bacterium starts forming like almost like poking its finger start extending towards this one, the other one start extending, and then they would eventually their extensions would connect. When they connect, they form like a tunnel. And what happens in the tunnel? One of these plasmids of this bacteria's DNA with the gene for resistance would move into this one. So this one that used to be harmful but not resistant can gain the competency to become resistant because of the transfer of material from this harmless one to this one. This process is called conjugation. So the end result is you have a group or a colony, they call them, of bacteria that are resistant. This one we don't have too much control over, but this one is the most one that we have control over because of the way we take antibiotics. I say prevention of antibiotic resistance ensures that we take all of our antibiotics as prescribed, not skipping doses or discontinuing treatment when we just feel better. And the other way to, again, is to make sure that we don't use antibiotics for things that don't require or for conditions that are not caused by bacteria. If you have a flu, make sure that you distinguish that your symptoms are caused by a virus and not a bacteria. Because sometimes you have like malaria, it feels like you are having a cold, but malaria is not caused by bacteria, and if a flu is not caused by bacteria. If you start taking antibiotics for it, it just wipes out all the other bacteria that where you had around, because the whole idea is bacteria don't know whether this other bacteria is harmful or this other one is harmful. It's just that when a lot of bacteria are around, they compete for the same space, they compete for food or resources, I'll just call that. But if you eliminate most of the ones that were having as competitors, the ones that used to be that can make somebody sick would have, again, more space, more resources to divide. I say do not use antibiotics for viral infections such as colds. Do not save unused antibiotics or take antibiotics prescribed for different infections. Look at this. They say each year in our country, 2 million people become infected with antibiotic-resistant bacteria. And to create a new antibiotics is not a simple thing. So if a set of bacteria type becomes resistant, it's a big challenge. You go to the hospital, you are sick, but unfortunately, they don't have a way to treat you because anything that they give you doesn't work. That can become very challenging. Now, let's shift and look at viruses. I said viruses are acellular. They don't have cells. You cannot say a virus cell, because when you use the word cell, it denotes the fact that it could divide, it could do all of the things that we say normal cells do. Viruses are acellular and sometimes described as obligate parasites. Obligate. Why? Because they need to get into another cell, into a host cell, for them to be able to divide, because they use that cell's machinery to divide. In terms of their structure, I said they usually have an outer protein coat called a capsid. And inside that capsid, they would have nucleic acid. Nucleic acid, you are talking about either DNA or RNA. So you have RNA viruses and DNA viruses. And in addition to this capsid, some viruses might have an envelope which would have some proteins poking out and carbohydrates poking out of them, like the coronavirus. The COVID, the viruses that cause COVID for us, we usually say, oh, you have the capsid has spike proteins on them, this red stuff on them. The monkeypox virus. So their structure, just remember this, they are non-living, usually referred to as virus particles, and they are obligate parasites because they require a host cell to invade it and be able to get into that cell for them to divide. What are some conditions caused by viruses? I think let look at this one here. Let's look at this one. It would show you an illustration, even if you don't hear any sound, but you can watch how the viruses would get into our cell. Here being the surface of our cell, and we have certain structures on our cell that could allow this virus particle to attach to them and see what happens as they get in. Let's watch this. Skip this verification. Turn down the volume. I think it's captioned so that you can see.
Virus attaches to host cell receptors by specific proteins on its surface called spikes. With the first mechanism, the envelope of the virus fuses with the plasma membrane of the host, and the virus is released into the cytoplasm. The capsid is removed by enzymes in the cell. In the second mechanism, the enveloped virus absorbs to the host cell by specific proteins on its surface, and the virus is taken in by endocytosis. The host cell plasma membrane surrounds the whole virus, forming a vesicle. The envelope of the virus then fuses with the plasma membrane of the vesicle, and the virus is released into the host cytoplasm. The capsid protein is then removed, and the nucleic acid of the virus is released. A naked virus that does not have an envelope also enters by endocytosis. There is no envelope present to fuse with the plasma membrane. After being engulfed, the virus is released from the endocytic vesicle. The nucleic acid then separates from the capsid.
So what we see here is that this virus can attach to receptors and get in here through a process that plants cells, our cells normally bring material in through endocytosis. And when inside the endocytosis, the capsid now is removed and the nucleic acid, RNA or DNA, becomes available. And it is this DNA that being available, the things that our cell use to make duplicates of it, they go there and they read the DNA, they make messenger RNA from it, and then make more particles, and so it goes. That's how it replicates. Examples again of some viruses are the ones that causes influenza, the ones that causes HIV, chickenpox, H1N1 for colds, rabies, Ebola. And the thing about viruses is, you think that only they attack only plants or animal cells? No. Viruses too, there are some that would attack bacteria. So bacteria themselves have issues with viruses. The ones that attack bacteria, they call them bacteriophages. Influenza, you are coughing, you are sneezing. Chickenpox, we showed like the measles, they're almost like that. They have these bubbles or these blister-looking structures on your skin. And then let's go even here. Now that we are talking about viruses, let's just segue here and talk about coronaviruses. That was a big challenge for us several years ago when a bunch of people were dying as a result of it. I said, what are coronaviruses? It's this group of viruses that causes diseases in other animals, including humans. They usually circulate in other animals like cats, rabbits, bats, and camels, and things. But sometimes, even though they are existing in these populations, they could have mutations that would make them, because we associate with some of these other animals like camels, if you live in the desert, they use them for transportation and things like that. So if birds hang out, these bats hang out close to camels, they can infect the camels. And when they infect the bats, they might even be living with these bats and not making the bats sick. But because we end up interacting with these animals, when they transfer to us, we can become sick. So if one of the things that you will notice is that there is a population of microbes that would stay with us and doesn't make us sick. But if they transfer from us and go to other animals, it can make those animals sick. This situation where you have pathogens like viruses moving from certain population, and which in that population they are not sick, but they transfer to a different population like us, and we start getting sick. They call this zoonotic transfer, viruses. So moving from one zone per se to another zone, and this will make us sick. The COVID vaccine, the COVID virus that we had even earlier forms that were different, like SARS, MERS, um, uh, but among these, COVID was the most dangerous. Now we are having H1N1 virus. I said symptoms vary in chickens. They cause upper respiratory tract. Most of these affect our respiratory tracts. There were times, if you see here, they said MERS, I think Middle East respiratory something virus, and then this one is South Asian respiratory something virus. All of them affecting our immune systems, and affecting our immune systems. And the symptoms we have is either we have colds, we have flu-like symptoms, and it's a big challenge in how we dealt with them. The coronavirus killed a bunch of people, and the bad thing about viruses is compared to bacteria, they mutate a lot. That means they change the order or the sequence of their nucleic acid a lot. Each time they divide, there's a chance that they're going to be some changes. And we had different variants of the COVID viruses as with time. But the good thing is we're able to come up with vaccines that kind of dealt with them. Some of the symptoms that people used to have, even with the H1N1 virus or these other ones, are similar to the ones that we have with the cold: runny nose, sore throat, headache, fever, cough, all of these things. And you see, if you have a sore throat, you can think that, oh, it is caused by a streptococcus, and start taking antibiotics. But it could be, if you had all of these other ones, it can end up being caused by a virus. But now they have a way to actually test to find out whether you are having a viral infection or whether you are having a bacterial infection. Some of the tests that they were doing, they had this immunological test where you have you rub your nose, and then you put it in this liquid and then drop it in this cassette that has like the pregnancy test, and then you look whether you have two lines appearing to show whether you are positive or not. And then if you want to confirm it to be more accurate, they do a molecular test where they would take a sample and then they send it to the lab. They call it a PCR test. And all of those would confirm whether indeed what you are having is a bacterial infection or a viral infection, and all of that. We will talk about that at some point later. Now, talking about some of these viruses, it begs the fact that we should also talk about transmission, how diseases are transmitted. Like these respiratory things that affect our airways and things that involve us having either runny nose, sneezing, and all of that. Sometimes they had asked us to take certain measures against COVID. They said you have to stay 6 feet away from other people. You have to wear masks and things like that. I don't know whether you ever ask, for example, we know, I will just talk about it, that when you wear masks, they said, oh, wear mask. What kind of mask? We said viruses, some of them have, point something nanometers. Meanwhile, bacteria is like a micrometer or two micron, or micrometer, you can call it same thing. So people were asking, should I wear a cloth mask or the one they say N95 mask? And then they were saying stay 6 feet from each other. What was all that? So to give a simple explanation on some of this, if you do not know already, when you sneeze, when there is no air, the estimation is that your sneeze droplets would go like about 6 feet or less than 6 feet away from you. So imagine that this person is sneezing here, and then because of the size of the droplets, then they can go around this, they'll go extend to around this much. So the deal is, if you were going to be standing 6 feet away from somebody who is sick and they sneeze, the sneeze will not go too far. But imagine that this person is sneezing with even a cloth mask. The cloth mask would be helpful, not that it would eliminate the viruses from passing through it, but it would reduce how far the amount of droplets that could go too far. But again, if you want to actually, if you are working in environments where you really want to make sure that the virus particles don't go through, there is an N95 mask. N95 is a brand, but the holes in the mask are small enough that the viruses cannot go through. So they were talking, wear mask or wear N95, wear regular mask. So all of these were measures that were being suggested or recommended for people to use. And this will also work if you have a regular cold or a regular flu, and that makes you have these symptoms of sneezing and all of that. If you wear something to cover your mouth, it will just reduce how far the droplets can go. And better yet, if you wear a N95 mask, you can work close by to somebody and then don't have, even when you speak, things will not go through. And then because we have this, we have our Center for Disease Control that was monitoring, they were monitoring trends of viral infection. This is the one that they were doing, for example, in 2021 when they were measuring COVID infections in the United States. Now that we don't have COVID, they're no longer measuring it, but now they're measuring what? But different kinds of flu that we have. So since I think sometime like in the ending of September, sometimes in September 2024, the flu season started. Up to now, they've been measuring the different kinds of viruses that are flu virus strands that are available. You can go to this link. You could go to the Center for Disease Control site to find out what kind of viruses are being reported in your area. Because when you go to the hospital and they take a sample from you to test for what you are sick with, if it reports positive for any of these viruses, they report it into a database that is recorded, transmitted to the Center for Disease Control, and then they tally it up to show representation. Now, for COVID vaccines, we said there are some links here, and if you want to check again, this is the link that goes for the Center for Disease Control that could lead you to some of these things. But in terms of how to deal with this stuff, we know that they have vaccines now that have been made for COVID. They have vaccines now that have been made for seasonal flu vaccines. One of the questions that we're going to ask is, okay, how do you decide what should be in the vaccine that you are making it because for the next flu season, sometimes when it is getting close, they would already make vaccines against the virus particles that they anticipate to be coming in the next flu season. In the next flu season, then the question is, how do they know to do that? We will talk about that later on. This one, I just mentioned here some of the general things that most of you should know in terms of how to protect yourself against whether it is the COVID vaccine, COVID virus or other flu viruses. Some of the things are very simple. They said, wash your hands, wear gloves, wear mask when you are close to other people. Don't stay with other people that are sick and all of those things. And make sure that you take the vaccines when they are available. And then if you have symptoms, make sure you test right to know what is it before you start taking any treatment. Don't stay in closed doors. If you have people that are sick, make sure that the area is ventilated. This is some of the way respiratory or diseases that are transmitted by droplets from how when we speak, they call it droplets infections. But Are there other ways in which pathogens are transmitted? Yes. If you take blood that is contaminated, so definitely you can pick up that contamination if the blood is tainted, if it has infection agents in it. People who do drugs using needles. If heroin addicts, you would see some of them have issues, not even just because of the heroin itself, but is that they use needles that are not clean, that are not sterile, and they end up having infections from these pathogens, whether it's viruses or bacteria. Sometimes pregnant mothers through the placenta could transmit infections to their developing babies. If you have body piercings, for example, again, it increases the chances of exposure to pathogens. Go to tattoo. If you have tattoo parlors, or in the process, if they use things that are not, again, sterile, you could have transmission that way. When you have intercourse, body fluids, you could have, again, transmission that way. So in this transmissions would involve things like viruses, things like bacteria. And now let's shift forward and talk about the ones that I said were neither bacteria or fungus or viruses, prions. I said prions, I say they are infectious proteins. When we have we have them, these proteins in our nervous tissue. But some weird things happen. They change shape and then infect neighboring ones to make us sick. Let's look at how how they believe this look. I'm surprised that we cannot get the sound, but let's watch. So this is a normal protein that we have. This is like our neuron. And these purple things are the proteins that are there that are normal. But look at what happens.
Prions are infectious agents that cause transmissible spongiform encephalopathies, TSEs, in animals. Examples of these diseases are scrapie in sheep, Creutzfeldt-Jakob disease in humans, and mad cow disease in cattle. A prion, which stands for proteinaceous infectious particle, is a protein that can alter the shape of other proteins. The normal protein, PRPC, shown here, is present on the surface of neurons in both vertebrates and invertebrates. This protein may be converted by the abnormal prion protein, PRPSC. The prion protein is called PRPSC. The SC meaning that it is associated with scrapie. This protein may cause the normal protein to misfold and assume the shape of the prion protein. With time, the prion proteins aggregate to form clumps that can interfere with normal brain function. The infectious prion protein is acquired when an individual eats the meat of an animal with the disease. The prion protein is not destroyed by cooking and can survive the digestive system and be absorbed into the bloodstream. It can then pass into the brain. The prion protein binds to the normal protein and induces it to misfold. The newly created prion then converts more normal proteins into the abnormal conformation. An alternative hypothesis is that prions activate enzymes that modify the normal proteins in the cell.
So there is a hypothesis that is suggesting how these proteins that are normal existing in our nervous system, what makes them to change their shape. One of the hypotheses was that when you eat meat that has these proteins, these infectious ones, these ones that are an altered form of it, they would go and merge with the normal proteins that we have in us and make the normal proteins that we have to change their shape. And when they change their shape, they go and do the neighboring ones. It's almost like going around and converting all the other normal proteins to change their shape. When they change their shape, in the change shaped form, they aggregate. And when they aggregate, they pile on the neurons, and the neurons cannot do their job properly. The other hypothesis is that when you get this material, you are getting some enzymes that will go and cause these proteins again to fold differently, to change their shapes. The ultimate thing is proteins that used to look normal, proteins that were normal, their shape is altered by something that we get. It infects humans. There's a group of if you eat like the brains of other animals, some people look at it as a delicacy that they even eat it raw. And when you eat this raw, it just bakes. If the thing is infected, you have a good chance of having it. And when it goes into your brain, it causes these issues. They call different things in different animals. In cows, they call it like mad cow disease. The cows lose balance. In humans, they call it Rachel Jacob disease. And then in sheep, they call it scrapies. In cats, they have a version of it. So in deer, they have a different version of it. The whole idea is it messes up their nervous system. Now, so basically, this first section that we just talked about, what did we talk about? We talked about microbes, talked about the different types, indicating what they do, and which ones, the ones that are pathogens that when we take them, they can make us sick. So imagine that now we know the things that the microorganisms that can make us sick. Let's look at what happens. What do we have to be able to defend ourselves should we be infected by these things? So the lymphatic system is what we'll talk about next. How we deal with this. I said it's a system similar to the circulatory system that is composed of organs, vessels, lymphatic vessels, certain tissues that protect us from infection and keep us healthy, maintaining that the fluid that we have is balanced throughout our body, meaning within the amount of fluids that we have in our blood vessels, the blood volume is right. And then the ones that we have surrounding the cells in our tissues, called the tissue fluid, is right. And if there's any excess in our tissue fluids, the rest of it would move into this lymphatic vessels, lymphatic system, and make sure that it's almost like a reservoir where if there is more liquid on the outside, like within the tissue, the reservoir picks up the excess liquid. If there is less liquid in the tissue, liquid moves from the system and fills up in and replenishes the ones in the surrounding tissue. So I say it consists of lymphatic vessels. The liquid, when I said it is not just, it has liquid which is called lymph. As you're going to see, it's made of water, salts, and it has the white blood cells and some proteins. And then it also has lymph nodes. It also has the thymus gland and the spleen. These are the components that make up the lymphatic system. But now we'll take each of these and see what it does. The lymphatic capillaries, what do? They absorb excess tissue fluid, and then they will return some of it to the bloodstream. The lymphatic capillaries, called the lacteals, they are smaller. When we talk about the capillaries, like in the blood vessels, we say capillaries, but here they're lacteals. Some of them would absorb what? Fat and some proteins. And then they also have a distribution of white blood cells called lymphocytes, with the lymph nodules, the lymph nodes having a bunch of them. It's almost like a garrison that has all of these white blood cells. What do? They help defend us against pathogens. So if you look at the components, if you start from here, this is our heart. Slide above our heart, we have this tissue here called the thymus gland. What does the thymus gland do? It is here that a kind of white blood cell goes to mature. So let's even just dial back a little bit. When you start here, where are all the cells in our blood made? They are made from the red bone marrow. The red bone marrow is the marrow that is found at the end of our long bones right here. It makes red blood cells, it makes white blood cells, it makes platelets. All of them are made here. But in terms of maturing, some of the white blood cells that are made here would move to the thymus gland to mature there. What are the maturing activities that are there? The white blood cells that go here, usually called T lymphocytes because of where they mature, the thymus, they go there and make our white blood cells to recognize cells that belong to us, recognize features that we have on our own cells, to make it such a way that they don't go to attack our own cells. Now, you would now see that when these white blood cells mature here, some of them now will get back into the lymphatic vessels. All of these green things that go like this are lymphatic vessels. Then we have this structure here called the spleen. The spleen is an interesting structure because it does what? When red blood cells, for example, get old or something is messed up with them, they go in the spleen here, they are broken down. The hemoglobin, the iron is taken out of the red blood cells and sent out from the spleen to be used to make more red blood cells. They have white blood cells stored in them. And so if pathogens that are circulating come across here, the white blood cells would destroy them. And then if you go here, we said these tonsils, these nodes here, a bunch of them in our groin, a bunch of them in our tonsils here. Because if you think about it, from our nose, from our mouth to our nose here, our throat is right here. So things that we eat, things that we breathe in, we mostly might have pathogens present in them. So we have a bunch of white blood cells staying here in our tonsils, anticipating if there is an infection, they could rise up and deal with it right away. In our groin area, yes, through our genitals, we have an entry that could have pathogens come in. And if the pathogen comes in, these starts going through our blood, our cells and things like that. All of these white blood cells there's many of them white blood cells in these nodules that are concentrated here to be able to go there to challenge them. We're talking about excess fluid. Look at this structure here. So if you look at your hand right here, they took this part here. This is a lacteal, part of the lymphatic vessels. And then you have tissue fluid that is here. But these are like tissue cells, like here. The space here, this is a space that is filled with liquid, and then you have a blood vessel here, a capillary here. So when our blood vessels exchange material will leave from here and go into this space and then into the cells, and then from the cells back into the tissue and back into the blood. So the idea is, if you have excess tissue fluid and the blood volume is right, where does that excess go? It goes back into here, into the lacteal vessels. So again, if you see here, all of these lacteal vessels, they come somewhere here, and then they would empty some of the liquid back here into our veins to go back into circulation. So you have this system that is almost like a parallel system to our circulatory system, but missing from what is being carried in the circulatory system. They don't have red blood cells. In terms of examining the vessels of the lymphatic system, they are very similar to those, or to the veins that we have. The veins, as we know, carry blood back to the heart. And to enable them do so, they depend on our skeletal muscles to press on them and move the blood up in the vessel. And to prevent backflow, when our skeletal muscles have contracted and pressed them, these vessels have valves of them, one-way valves. So imagine that, just like veins, they have valves. So when you have skeletal muscle pressing on them, they would press and liquid would move to this section. And even when the muscles contract, the liquid doesn't flow back because of these valves that are there. And then if you look, I would move here and go to, when we look at the various organs, the various structures that are involved in this lymphatic system, some of them I started mentioning. We can classify them whether it's primary or secondary. Those that are considered primary would be the red bone marrow that is responsible for making the blood cells, and the thymus gland that I said some of the blood cells go to mature there. So you have, first, just to point out, yes, the red blood cells, the red bone marrow makes blood cells. Those that go to mature at the thymus gland are called T lymphocytes. Some of them do stay and mature in the red blood cells. Those will be called B lymphocytes. Now, the next thing is we have secondary structures. Well, I was mentioning lymph nodes and the spleen. The lymph nodes, again, they store a lot of white blood cells. Talked about the spleen, it stores white blood cells. It cleans the blood. It has red blood cells that are bad. It recycles them. I put a slide here, which you might not have on your current deck, to be able to talk again a little bit more about how cells produce in the cells that are produced in the red bone marrow. We said in our red bone marrow, we have multipotent cells. Multipotent. It means that these cells are stem cells that are capable of dividing to form different cell types. We say if you need red blood cells, for example, there's a substance produced by our kidneys called erythropoietin that would stimulate these red bone marrow stem cells to divide. When they divide, so imagine that there was one here, and then it gains stimulation to produce red blood cells. It will divide, and one of them would remain in this state, the multipotent stem cell, and then the other one will go and become a myeloid stem cells, like starts saying it's like developing, going to start making this, differentiating or changing into a cell that would ultimately form a red blood cell. And then the erythropoietin would come here and make this divided cell that originated from here to now go now and make the red blood cell. Different things can stimulate this one, but its ultimate came from here. Different things could stimulate it to produce but platelets. Others can stimulate it to form some white blood cell like mast cells. Others will stimulate it to form some other type here. So these ones will go here form mast cells. This one will form another kind of cell called myoblast, that with other further instructions again would make it grow to become this other cell, white blood cell types, basophils, neutrophils, eosinophils, monocytes. And then these monocytes, again, something would stimulate them to become this. Again, where do all of these cells came from? They came from this one that divided and now was stimulated based by different substances depending on the need of the body to produce different kinds of products, cell types, platelets. Sometimes this one, if we needed but like lymphocytes, lymph cells that exist in the lymphocytes. If we needed, our bodies needed but lymphocytes, this one would again divide. Then the factors that will stimulate it to make up to grow like a mother cell that would produce, when divided again and stimulated, it would go to produce these different kinds of lymphocytes. So the point here is the red bone marrow is super crucial. These are the cell types, as you're going to see, that will be involved in fighting, defending our body. The platelets, which are cell fragments, these basophils, neutrophils, eosinophils, monocytes, macrophages, dendritic cells, T lymphocytes, B lymphocytes. We will show in the next few slides how all of these players here are involved in making us defend our bodies from disease. You'll see here again, I put this as a different slide again, showing that right from the red bone marrow, you could go now to have this, they call it hematopoietic. Hemato is like how to deal with blood. Stem cell means that it has the characteristics of growing to become different cell types. And those is dependent on this various chemicals or factors that are provided by the body to stimulate it to differentiate. I use the word differentiate, means growing with particular features, right? So it can differentiate to produce red blood cells. Again, this is just a different notation of illustrating exactly the same thing. And then in the next slide here, I went and I put the notes, summarized the notes again of some of these various organs, like the red bone marrow, I said again, site for blood cell production. The other thing to note about the red bone marrows is that more bones in children have red bone marrow because, again, they need to grow, they need to develop, and they need to develop more of these cells for the cells of the blood. The thymus gland, as we're also going to see, in children, it is larger in children because, well, children are still growing and they need to make more white blood cells to be able to go there and be familiar and be trained to mature there and be familiar with what is self, to differentiate it of features that would be not belonging to the developing baby. I said it is largest in children and shrinks as we age, because as we are aging, we already have a lot of white blood cells which recognize our features, the features or the characteristics on our cells. Now, and then I mentioned the secondary lymphatic organs, the lymph nodes and the spleen, what they do. Now, let's see now what each of these cells do in terms of defending the body, which we can group them into a first and second line of defense. We call it innate. We are born with this. We are born with these features to defend ourselves. And then we have acquired. This one is where our cells actually learn, or they need to be presented with something to learn about it and know how to defend against it. I think we should take a break here and take like a bio break for about 5 minutes before we come back and talk about the section that we'll talk nonstop until we finish.
Videos, go to www.makemegenius.com. Oh, yeah, it is free.
Hey there, I heard you were sick. How are you now? I got you some flowers. Oh my God, bless you. Looks like your immune system is working really hard. Don't you know about the immune system? They are the most amazing army ever, and they keep fighting the toughest battle all the time. Don't get so excited. I'll tell you all about this amazing defense which our body has against bacteria and viruses. Let me first show you the world around you with a microscope. You see, our world is filled with lots of germs, yet we're usually healthy thanks to our immune system. The immune system is an army of organs, tissues, and cells that work together to keep you safe from these germs. These germs, viruses, bacteria, fungi, and parasites try to enter your body all the time. The immune system uses many tricks and weapons to keep them away and kill them. Your body has 3 levels of defense. The first are your skin and mucous membranes. Second is swelling or fever. And the third is the internal immune system. At the first level, your skin and the mucous membranes of your nose are the first barrier for these germs. It's not easy to get inside your body, covered with a waterproof, germ-proof, self-healing skin. Oh, take it easy. In case your skin is getting cut through or a bee stings you, then the second level of defense comes for help. The red swelling and your fever are all ways of the immune system to limit your damage.
Sometimes you get a fever, your body temperature goes up, and viruses are killed due to high temperature. So a fever can protect you from viruses.
So, what are the first and second level defenses of your immune system army? Very good! But it's not all about defense. Even if the first and second level defense fails, and a germ is able to get inside your body, then the third level of defense for your immune system starts working. When a virus invades your body, it tries to take your healthy cells and makes more copies of itself. This makes you feel sick. We must stop it. Your third level immune system follows a 3-point attack plan. It identifies the invaders, kills them, and then remembers them for future battles. So here is how it happens in the body. The first step is to identify them. Special cells called macrophages do this work. They surround the enemy and raise a flag to call the deadly attacking battalions. This flag indicates an antigen. This means there's an enemy cell which needs to be destroyed. Your weapons will be of no use. To kill the germs, the immune system has a battalion of T cells, B cells, and killer K cells, which work together to kill these cells. Do you want to know more about these? Then there are M cells, who remember the enemy viruses so that if they come next time, they are killed immediately. Do you want to know more about these? T cells are helper cells that recognize the antigen flag on the macrophage. They call out to their friends, the B cells. The B cells produce Y-shaped antibodies. These antibodies attach to the antigen and tag them for destruction. Kind of like a game of tag or catch that you play with your friends. Now it's easy for the killer K cells to destroy all the germs because they have been tagged by the T and B cells. Then comes the K cells. They will destroy all the germs. That was a lot of stuff to understand. Do you remember who are the fighters of our immune system army? Yes, yes! There's still work left to do for the immune system. Special memory cells, called M cells, carry the memory of an attack even after the victory. This makes our immune system better prepared for the next attack. Your immune system works hard to keep you healthy. Would you like to know how you can help it? Wash your hands frequently. Most germs enter your body through your hands. Get your vaccine injections on time. The vaccines help strengthen your immune systems against many dangerous diseases. Eat healthy, sleep nicely, and exercise regularly, so your immune system has less to do.
So you're going to...
So this is a very basic intro that you could do like for fifth graders to talk about the immune system in a very simple overview. So we're going to look at the first and the second lines of defense, which are usually innate. We are born with them. And then we'll look again the third line of defense, which would be active and passive immunity. If we go here, we said in terms of characteristics, these innate defenses are non-specific. So they just look at something, understand that something is coming, invading the body, and they just know that it's different. It doesn't belong here. And they're trying to prevent invasion, or if they do, they want to how to deal with it. The first line of defense is usually the barriers that prevent material from coming in to the body. We can call it the physical barriers and the chemical barriers. In terms of physical barriers, we have the skin. Our skin has areas that will prevent, even though we have some bacteria on our skin, it also still prevents them from coming in. And then we have the tears that we have in when we cry, or we don't even have to cry. You know, when you blink your eyes, you have this tear glands produce this liquid that keeps washing their eyeballs. And the tears have lysozyme, an enzyme which, if some pathogens want to click on them, they destroy. And then in terms of the urine, when we pee, we flush out microbes. Is there any microbe that wants to stay within the part that is exposing almost close to the outside? We keep flushing things out. The mucous membranes that line our respiratory tract. If you look from when we are breathing through our nose, right through our trachea, into our lungs, the walls of that opening, because it's almost like a continuous tube from our nose, is open right into our lungs. The lining of that tube is made up of mucus, below which are cells that secrete the mucus. And they also have these hair-like structures called the mucus. They have cilia on them. If foreign stuff wants to get in, it gets trapped by the mucus, and then the cilia whips it up and brings it out so that you could cough it out and blow it out. Our digestive tract also has mucus. Our reproductive tract, our urinary tract, all these ones, they have things that would gum up any microbes to make sure that they can be flushed out. Then the other part I was talking about called resident bacteria. Sometimes they will just call it general because there are different kinds. They call it normal flora that stays with our on top of our skin or inside our stomachs, for example. It makes that when the other bacteria come in, it competes for things and doesn't even give them chance to proliferate, to grow. I was talking about our stomach. If you look in our stomach, what is the pH in our stomach? pH of 2. The pH of 2 makes it such that if you could eat your salad, even though it has some bugs on there. When that pH of 2 and some of those bacteria that are not used to staying in that low pH conditions, they get into our stomach, they die. Our skin, I was mentioning, it produces, there's some oils that come from oil glands underneath in our dermis area that come up and into the surface are called sebaceous glands, produce sebum. These oil things, these secretions, they have material that would make some pathogens not to be able to grow. And then the vagina has a pH of also a low pH again, so that any bacteria that wants to get in through that way. So between this combination of physical barriers, the mucus stuff, the chemical stuff, it limits or prevents a bunch of pathogens from getting in. So sometimes we can do a good job doing this prevention, but some still get in, or sometimes accidents happen. Let's say, for example, somebody is doing some kind of thing, and then you poke your hand. When you poke your hand, you have a situation where blood starts coming out, and then you hold it tight. It stops bleeding. But before it does bleeding, when you just, even what you were using that poked you, there might have been some, it was not sterile, there might have been some pathogens on there. The green things that are illustrated are shown here, for example, are pathogens. Now you poke your skin here, all of these pathogens start finding their way in here, and blood too is coming out, because this is your blood vessel here, this is the skin part, this is the tissue here. Now, if you get this far, the blood starts coming out, you hold it, the blood stops. But look at what is happening to this, to these green things. Now you have some cells here. This is where we start talking about some of those cells that I said were the white blood cells that are produced from our bone marrow. There are some cells here called mast cells. They would produce chemicals. If they notice like injury to the skin, the injured cells here and the mast cells, they will produce some chemicals called histamines. What do these chemicals do? What they do is they make the blood, when your blood vessels sense the presence of these chemicals, they open up, they dilate, they get bigger. Why do they get bigger? They get bigger, or when they get bigger, what happens? More blood is able to come to that area. When more blood comes to that area, because the blood is coming from within, it is at a slightly higher temperature. So at a higher temperature here, it makes this area to feel warmer. And sometimes just the variance in increase in temperature is able to mess up some pathogens. So it's almost like non-specific. Just the increased temperature probably could mess things up for some pathogens. The other thing that it will also do, it starts, as it's coming, it is bringing some other cells, some other white blood cells called monocytes and neutrophils to come out from the blood and get into the area. And these would change to macrophages, and these macrophages, they have a capability of doing what? Of engulfing stuff, phagocyting stuff, doing phagocytosis. They would engulf these foreign things, bring it into themselves and mess them up and destroy them. Meanwhile, I mentioned that when we press the area, it stops the blood from floating, from bleeding. And that stoppage is caused because what? You have this clotting process that happens with fibrinogen gets converted to fibrin to form fibers to clot it. Let's watch a quick video here that illustrates the system.
Inflammatory responses are one of the non-specific defense mechanisms that protect us from diseases. Now, let's see how an inflammatory response is produced. Pathogens may get into our body through a wound on the skin. In the wound, damaged cells release certain chemicals. These chemicals cause dilation of nearby arterioles.
Histamine.
Thus, blood flow to the capillaries around the wound increases. The wound becomes red and hot. The chemicals also increase the permeability of the capillaries. Thus, more phagocytes come out of the capillaries to engulf and digest the pathogens. As more tissue fluid is formed and accumulated at the wound becomes swollen. This sequence of events is called the inflammatory response.
So again, this is non-discriminatory. They just notice that something has happened. Strange stuff is coming in that is not supposed to be there. More blood flow is caused to happen in the area. Because more blood comes there, the area feels warm, it feels swollen. And then when these blood vessels are responding, opening up, they make them to much more permeable because it makes these cells, these white blood cells, to be able to come out. When they come out, they are going for what? Looking for all of these things that are foreign to be able to destroy them. So you imagine that when this happens, many times when you poke your hand and it stops bleeding, everything heals back and everything goes to normal. You don't have an excessive infection going on. It gets resolved in a couple of days. So you can see that just by this process called the inflammatory response, involving just this set of actors, you are able to prevent something more crazy, more extensive happening. So it's successful in this case. Now, if you go now and said, anyway, this notes what I just described here is found right here. And then, but what happens if you end up really having an infection where we normally say we have an infection, where that response did not resolve it, out of a sudden, the first day they place the kid swollen, and then it keeps getting swollen, and then pus forming, and then it's not healing. Then you say, hey, maybe this stuff here, I need to go actually to the hospital to check it out. So let's look at the next step of what happens to be able to deal with this process. So this second line, we had the first barrier had a barrier system that also had chemicals dealing with it. And then the second system, now we had some white blood cells called the inflammatory response. We had redness in the area, we have heat, and we have swelling. We might have pain in the area, but we have the surrounding cells producing these chemicals. These chemicals now forming dilations. Then we have cells that come now and engulf bacteria. That is the step. Now let's go now to the next step. And to go to the next step, I'll kind of set up again, tease up here. You saw this slide when we're talking about the white blood cells that are formed from the bone marrow. I said again, we have lymphocytes. And then we have these ones, neutrophils, eosinophils, and basophils. These ones have granules in them. They call them granulocytes. These two here, monocytes and lymphocytes, they don't have granules in them. You will start seeing how these ones come to play. I put again another picture here to tie back from what I showed you before. You have the bone marrow, the stem cells producing this set of cells. Now you're going to see how these come to play and how these ones come to play. I'm going to skip this one here and go right here. So in this line of defense, there are some proteins. still in the second line of defense, besides those besides that inflammatory response to mention, there are also some proteins called complement proteins that exist in the plasma. What they do is slightly different. They would go and bind onto the green pathogens and make holes in them and compromise them to burst open. And then we have some other different kinds of proteins that if a cell is infected by a virus, it produces these proteins called interferons to warn neighboring cells that I have been infected. You prepare yourself in such a way that before the virus comes to infect you, you have some of these complement proteins and other things to be able to attack it upon infection. So this is part of the second line of defense, which was the inflammation. Now, if we're going now to the third line of defense, it now has to do with all of these cells that I had mentioned to you here, which are these neutrophils and all of these other cells. We'll take a look at it and what happens. But generally speaking, I would say if you look at after you have the barrier system has not worked, the protective system has not worked, now the infection has gotten into your cell. What happens is you would have cells that would engulf this pathogen, and when they engulf the pathogen, they take some of the pieces of the pathogen and project them out and put them out on their surface. And when that pieces of pathogens are projected on their surface, these cells, some other white blood cells, are able to recognize that there was a pathogen with some characteristics, and then they know now how to go and participate in the fight. So let's take a look at this other one here. So it ties both systems together. Let's take a look.
Every day, we're exposed to countless pathogens, but don't get sick every time. So how does the body recognize and fight these pathogens?
I wanted this to make this bigger, sorry.
Every day, we're exposed to countless pathogens, but don't get sick every time. So how does the body recognize and fight these pathogens? Let's discuss this step by step. The first physical line of defense is provided by the skin and mucous membranes. In the airways, mucus, ciliary motion, and coughing are important defense mechanisms. In addition, bodily secretions also protect us from pathogens. For example, saliva contains lysozyme, which can destroy the cell wall of gram-positive bacteria. If we take a look at the stomach, gastric acid creates a hostile environment for most bacteria. Also, the normal flora residing on the skin and mucous membranes displace and fight pathogens. If the pathogen ends up breaching these barriers, the body depends on further active defense mechanisms of the immune system. First, the cells involved in innate immunity become active. Certain stimuli, such as tissue injury, trigger an inflammatory reaction. As a result, leukocytes, especially granulocytes and macrophages, are attracted to the site of damage. They leave the adjacent blood cells and penetrate into the damaged tissue. The body recognizes certain specific repeating patterns present on many pathogens as foreign. These include, for example, lipopolysaccharides, which are located on the outer membrane of gram-negative bacteria. These characteristics are also called pathogen-associated molecular patterns, in short, PAMPs. because they're commonly shared between different pathogen subtypes, they're considered nonspecific. The macrophages and granulocytes attracted by the inflammation recognize the PAMPs of pathogens through pattern recognition receptors, in short, PRR. The granulocytes then secrete a toxic substance to fight against the invading bacteria. The macrophages engulf the bacteria, or in other words, phagocytose them. In addition, PAMPs activate the complement system. This system consists of many different proteins that initially circulate in the blood and tissue in an inactive state. These proteins are activated by pathogens, leading to a cascade of reactions. This leads to three effects. First, more leukocytes are attracted to the site, which stimulates the inflammation. Second, the complement components mark the pathogens, facilitating phagocytosis, which is required for some pathogens. Third, the complement cascade leads to the formation of the membrane attack complex, in short, MAC, which produces pores that especially kill gram-negative bacteria. As well as the innate immune system, the adaptive immune system is also activated. Its reactions start at a later stage, and the response is initially slower. Through its high degree of specificity, the adaptive immune system can efficiently fight against the invading pathogen, preventing it from spreading.
So I put many of these videos here for you to see because it makes it much more illustrative of what is going on if you see the sequence and then you read the story to illustrate it. And then I said the third line of defense is called adaptive defenses. It is much more specific. It means it targets specific pathogens, and it always happens when the first and the second line have failed. It looks at structures that are on the pathogens, whether it is the virus or whether it's in the bacteria. It identifies those and specifically hones on those characteristics that are associated with the pathogen. It also helps us to deal with cancer cells. Remember those lymphocytes? I said the T lymphocytes, they go to our body to be trained in the thymus about the characteristics that our cells have. They know when the characteristics that a normal cell would have. When a normal cell becomes cancerous, means it is dividing without control, it also assumes certain characteristics that are different than when the cell used to be normal. This third line of defense are able to recognize those and also deal with those cancer cells. The reason sometimes cancer kills people is because the cancer cells divide faster than the immune system can deal with them. There are many times which we have these abnormal cancer cells that come up, but our immune system goes and deals with them before they become an issue for us. So who are the players in this third line of defense? They are those cells called lymphocytes. They are the B lymphocytes, the white blood cells that mature in the red bone marrow. Hence, they are called B lymphocytes, and T lymphocytes, those that mature in the thymus gland. So we could describe these two, into two sections. Some of them deals with cells that go and target the pathogen and destroys it directly. The other one has to deal with antibodies that go and tag on the pathogen for other cells to come and deal with it. I say in antibody-mediated immunity, also called humoral immunity, B cells would produce antibodies. There are some white blood cells, as you're going to see, that cannot recognize. It's almost like they are blinded to the characteristics of the pathogen. But the antibodies go and identify the pathogens and tag them. When they go and tag the pathogen, then those destructive cells are able to visualize that, oh, they have tagged this thing, it means it's meant for destruction. It's almost like when the military goes to certain areas in a war zone to identify buildings that they should bomb, they go and put some tags on them, because the military, the defense aircraft or the military aircrafts with bombs cannot differentiate which building is supposed to be bombed or which one structure is supposed to be destroyed. But with the help of specific tags that identify those buildings, those bombers are able to go there and target them and destroy them. So antibodies serve like the tags that identify things that need to be destroyed. And then we have killer cells that go now and try to go now to destroy them. So the B lymphocytes produce antibodies, which are required for tagging. The T lymphocytes have a subset of them that go now and do the killing. So I said here in there, I said there are subtypes. We have B cells, which are the B lymphocytes. When the infection comes, the variety of B cells will try to see which one will go and connect, identify some of the characteristics on that pathogen. The one that fits and recognizes it, that particular B cell divides. When it divides into two cell types. It will divide into a plasma cell and a memory cell. The memory cell just says, oh, this is I have seen this before. And then they will stay and don't do anything. But the plasma cells go now to shed out, start producing, they divide and start producing a lot of antibodies, the same kind of antibodies, which now would go around in the system and identify anything that has that particular structure would be destroyed. I said the T cells are three types: cytotoxic T cells, helper cells, and memory cells. So let's take a look here. So if B cells, we have different kinds of B cells. Different because they have receptors on them, structures on them. So, to show the variety, they kind of illustrated them here. Look at this B cell. It almost have like a cup-shaped U structure on its surface. This one has a V-shaped structure on its surface. This one has like a half square or three-quarter square structure on its cell. The whole idea is we have a variety of B cells with different receptors on them. These receptors would recognize some of the structures or the antigens on particular pathogens. So let's look at this one. If you look at these ones, look slightly round, the antigens on these pathogens are slightly round. If they don't fit properly here, they will not fit properly here either. But if you look at them, they would from these three, they would fit better on this particular B cell. So when they fit particularly on this B cell, this particular one would replicate, means it will divide, more of it will divide. When it divides, it forms two cell types. One of these one forms plasma cells, and then the other ones forms the ones that more of this same kind. These ones will stay like memory cells, but this one you see now, these structures are not longer on them. When it produces this structure, it shades them out. These structures are the antibodies that they produce. So these B plasma cells that come from the activation of this particular one would be producing antibodies, because these antibodies will go around now in our system between our tissues, and each time they see these ones, they will bind it up like this. They will bind it up like this. When they bind it up like this, the cytotoxic T cells would recognize this complex of antigen plus antibody, and then they'll come and destroy it. So basically, they will mop out all of these green antigens from the infection. Pathogen with characteristics or antigens recognized by a particular B cell. The recognition is being that it has receptors that can bind it. When that recognition is done with this one, we say this B cell is been activated. So these ones will not be dividing. These ones will go now and divide. When they divide, they start making more of these cells, but they go into two groups, a plasma cell and a memory cell. The memory cell has the same structure, almost like the original parent B cell that was activated. Meanwhile, the plasma version of the replicated cell starts shedding these structures called antibodies and making many of these antibodies. It allows these antibodies to go into the tissue, go into the blood, to be able to go now and bind the pathogen represented here in green. When they bind the pathogen, this complex of pathogen plus antibody enables the killer cells to come and now destroy it, the T lymphocytes to come and destroy it. After all these green things have been destroyed, what happens to these? These plasma cells that were producing antibodies get destroyed themselves through a process called apoptosis, called programmed cell death. They look like, okay, there is no more need for me to produce more antibodies because those that were, all the pathogens have been destroyed. So they self-destroy. Meanwhile, this particular group of memory cells now, we have a good population. They were, in this case, just imagine that they were just like one or two or few of them. But now, because they were involved in this encounter of this pathogen, many of them remain in our tissue. Such that if a secondary infection comes, these ones would be easy. There are many of them now that they can easily go and now bind and then do the same process all over again.
B-cell clonal selection theory. Immune response consists of multiple levels of reaction to a potential pathogen in the body. The purpose of the adaptive response is to recognize, respond to, and then eliminate specific particles entering the body, called antigens. An antigen is anything that may initiate an immune response, such as foreign proteins or fragments of the cells. The adaptive immune response is carried out by white blood cells, called T cells and B cells. In this video, we'll examine the role of the B cells. B cells are responsible for production of antibodies that target specific antigens present in the tissue fluids of the body. The B cell response is specific because each B cell contains a unique B cell receptor, or BCR. Each B cell receptor recognizes a specific antigen. Because antigens are often found in the tissue fluids of the body, they eventually make their way to the lymph nodes or spleen, where a large number of inactive B cells reside. If one of the B cell receptors recognizes an antigen, then that B cell is activated. Once activated and exposed to chemical signals called cytokines, the selected B cells undergo clonal expansion. Clonal expansion basically means that the activated B cells divide continuously, producing exact replicates or clones of themselves. Most of these B cell clones develop into plasma cells. Each plasma cell is specialized in the mass production of lots of identical antibodies, which are very specific to the antigen that they target. Because each of the B cell clones is an exact copy of the activated B cell, the clonal expansion process produces an army of identical B cells. Each plasma cell in this army produces large amounts of antigen-specific antibodies. These antibodies help tag the antigens for destruction by a variety of mechanisms. For example, once tagged by an antibody, some pathogens are attacked by the complement system, which lyse cells. Other antigens are attacked by macrophages or other phagocytic cells. While most of the B cells become plasma cells, a few are held in reserve as memory B cells, which are active in long-term immunity. Like the original inactive B cells, memory B cells reside in lymphatic tissues of the body. Their job is to protect the body should it be exposed to the same antigen in the future. If the exact same antigen enters the system again, memory B cells react and mobilize. They quickly divide and give rise to more plasma cells capable of rapidly producing the correct type of antibody.
So the goal here is we have exists a variety of B cells in our body waiting for any different type of antigen brought in by a pathogen. And once the right one, it takes some time to be able to find out which is the right of B cell that would identify it. But when the right B cell is able to identify the pathogen, it goes that replication, clonal expansion, and the rest of it. And all of that again is indicated here. But one of the questions is always, what happens when the infection is gone? When the infection is gone, the plasma cells that were producing antibodies self-destroy. The memory cells stay behind, waiting for the next time around. I just kind of put one slide here regarding what an antibody looks like. An antibody is a Y-shaped structure. Most of the parts, like the part of the stem of it, is the same, but it is the Y, the two arms, the ends of it, that they call them the variable region, because we have different varieties of these variable region shapes, which are meant to give many more selections to be able to identify and bind specific antigens. So you would hear, I put this here for us to see in terms of different kinds of antibodies that are produced by some B cells. And we have different ones that do different things. I say like IgM, the IG stands for immunoglobulin. I say the main type that is in circulation and is able to help the baby. It crosses the placenta of the pregnant mother to be able to help the developing fetus in case any infections want to cross abroad. Across to infect the baby. IgM says the main type found in circulation, the largest amount. Again, the first that is formed when the baby's own immune system is forming. We say again, it would produce its own antibodies that will go and tag a pathogen and then allow for complement proteins to go and now cause holes and compromise the antigen. The IgA, this one's again secretions found in milk and saliva. And then this one, the IgD, I said is the most kind of the common antibody that is on the surface of immature B cells. And then the IgE, this is the one that is usually found in the ones that are responsible during an allergic response. Then let's talk about the T cell part of this. I said T cells, they are involved in cell-mediated immunity, but they go and attack viruses and cancer cells. Their origin again comes from the red bone marrow called T cells because they mature in the thymus gland. We have one set of them that are called T helper cells. We have another one that are called T cytotoxic cells, and then we will have T memory cells as well. Their action is again slightly similar to the B cells, but also different. We have different ones that have different kinds of T cells that have different receptors on them. The ones that have receptors that will correspond to the particular antigen will go now and they will bind to it, or sometimes they will take up. First of all, they are blinded. They are blinded to the antigen, but when a macrophage takes up, engulfs, endocytoses the pathogen, it would show on its surface here if an antigen that is from the pathogen. When it shows on its surface, these particular T cell types, the one that will bind to it would be the one that will be activated in a similar manner as the one that was going on with the B cells. So it would produce more of the cytotoxic T cells, but in concert also produce some that are memory T cells. The cytotoxic T ones are the ones that go and destroy the bound, the bound that go and recognize, and anything that binds to the pathogen would be destroyed. And then the other ones would again stay and be memory cells waiting for the next encounter. And you could look at that, the clonal expansion of this one as well. Again, it targets viruses and cancer cells.
When a virus infects a cell and synthesizes viral proteins, some of these proteins are degraded to peptide fragments. These peptide fragments form a complex with class 1 MHCs and are displayed on the surface of the infected cell. Cytotoxic T cells interact with the virus-infected cells by recognizing both the viral antigen and the class 1 MHC. The cytotoxic T cell then releases perforin molecules which create pores in the plasma membrane. Enzymes called granzymes are delivered into the pore, which cause the cell to undergo apoptosis. Cytotoxic T cells only bind to foreign proteins presented on the surface via the class 1 MHCs. Self-proteins are not recognized. Once the cytotoxic T cells have released their perforins and granzymes, they detach from the infected cell and move on to other target cells.
Now let's look at the one that was presenting, the helper cells that do the presentation.
T-cell clonal expansion. The immune response consists of multiple levels of reaction to a potential pathogen in the body. The purpose of adaptive responses is to recognize, respond to, and then eliminate specific pathogens in the body based on proteins found on their surfaces, which are called antigens. In this video, we'll examine the response of T cells, which are immune cells that play a very important role as the link between the nonspecific actions of the innate immune response and the pathogen-specific adaptive response. When a pathogen, such as this virus, enters the body, it initially encounters cells associated with the nonspecific defenses, such as dendritic cells. One of the more common dendritic cells is the macrophage, which destroys pathogens by ingesting them during a process called phagocytosis. Once the macrophage has destroyed the pathogen, the virus in our example, a small piece of the pathogen is displayed by a specific receptor on the surface of the cell. This receptor is called a major histocompatibility receptor or MHC1. The piece of the pathogen that is attached to the MHC1 is the antigen we mentioned earlier. This antigen will invoke a specific immune response by the body. The macrophage is now referred to as an antigen-presenting cell, or APC. Like the B cells of the immune system, each T cell has a unique surface receptor. In the case of T cells, this receptor is called a T cell receptor, or TCR. Each of these receptors will typically recognize only one type of antigen. Unlike B cells, however, the T cell receptors are only able to recognize antigens that are attached to the MHC1 marker of antigen-presenting cells. The type of cell that makes this connection is a helper T cell. It plays an important role in making the connection between the nonspecific actions of dendritic cells and the clonal expansion of T and B cells. The identification of an antigen-presenting cell by a helper T cell causes that helper T cell to become activated. The helper T cell releases chemical signals called cytokines, which cause the activation of B cells in the lymphatic tissues of the body. At the same time, the helper T cell undergoes clonal expansion. Clonal expansion basically means that the activated T cell divides continuously, producing exact replicates or clones of itself. These clones, now called cytotoxic T cells, are designed to target other antigen-presenting cells in the body. Once the cytotoxic T cell recognizes an antigen-presenting cell, it releases chemicals, such as perforin, that result in the death of the infected cell. The T cell response targets infected cells of the body, while B cells recognize and destroy any free antigens, such as viruses, in the tissue fluids. While the T cell response is underway, a few of the T cell clones are held in reserve as memory T cells. Their job is to protect the body should it be exposed to the same antigen in the future. If the same antigen enters the system again, the body can quickly activate specific memory T cells, which then divide and target infected cells. Once the infection has been cleared and the antigen is no longer present in the tissue fluids, the cytotoxic T cells undergo apoptosis, thus protecting the body from overabundance of cell-destroying cytotoxic T cells.
So we have the B cells and the T cells that identify specific pathogens or antigens for destruction. And when they have gone through the process, they have memory cells, whether they're memory B cells or memory T cells. And because of that memory cells reservoir that are there, anytime the secondary, the same kind of pathogen wants to come and destroy, invades the body again, there are many of them that could go and identify that pathogen early enough and start replicating fast enough to make sure that if the pathogen is trying to divide, because they have many of the activated cells coming into play now, they would overwhelm, whether producing the antibodies in the sense of the B cells or the helper cells in the case of the T cells, to be able to overwhelm the virus and destroy it. Any question between the B and the T cells? Is anybody still there? Are you guys still there, or everybody left?
Yeah. Yes.
Any question?
I have a question about the exam.
No, we'll talk about the exam later on.
Okay.
I can tell you about the exam that this B cell stuff is not, and the T cell is not in the exam. So, but we'll talk about the exam questions later. But I want us to find out, I'm just trying to find out whether we have any questions about the players in the ways our bodies defend. So This is where the body defense on specific is more targeted. And I said there were B cells and T cells involved. And I talked about the B cells. We talked about where they came from. And then we said in terms of the process that we have different kinds of B cells that differ because of the kind of receptors they have on their surface. With those things that are those receptors, those things that are poking on their surface, and their tips are slightly different. That's the variance. And we said just generally that when you, depending on the antigen, because the antigens don't have the same kind of receptors, specific properties, the right B cell receptor that matches that antigen, and that match would mean is being able to bind to that antigen, that particular type of B cell now starts dividing, making more of itself. In that process, they call clonal expansion. A clone is just basically a bunch of the same kind of cells. So it's almost like you have B cells, each wearing different kinds of caps. Some have red caps, some have blue caps, green caps, and then out of a sudden there is a pathogen. Let's say red matches red. Then the red B cell, the B cell with the red cap now goes and recognizes, oh, this is the red antigen, and then it binds to it. Then out of a sudden, that red one B cell now starts making more of the, more of the same kind of B cells now with red caps. That's the clonal expansion. But meanwhile, the other B cells with green, blue, white caps, nothing happened because they didn't have anything. They didn't match that particular antigen or that particular pathogen. And then when these B cells do this clonal expansion, remember I was using the cap analogy, and let's say they now divide to B cells that divide and have their caps on, and then the B cells that shed their caps, just other groups of B cells that keeps releasing their caps, which are the antibodies, keeps releasing red caps. And the red caps now already have the shape of going to bind to the red pathogen. And that antibody pathogen association now is eventually destroyed. T cells, something very similar but slightly different. The T cells, they do not bind, they do not go directly and bind to the pathogen. Instead, they have T cells that would engulf, would bring in the pathogen into itself, and then the piece of that pathogen, its stock act is almost like it rates a flag that has a piece of the pathogen on its surface. And it is only until then the variety of T cells that have different receptors too, the appropriate one that matches, that goes and matches to that flag and recognizes and binds, goes the same kind of clonal expansion to make more of that kind. And then the more of those kind now go now and bind to more of the macrophages with those things sticking. And then that association makes them to send chemicals that will go and destroy them. And some of the ones that had matched the cap would stay like memory cells. So I'm hoping that these videos would enable you to understand them better. And then we would call all of these cells that were in the process of engulfing and then presenting the piece of the pathogen on their cells, on their surface, helper cells. Because how were they helping in the process? They were bringing in the pathogen and then taking a piece of it and making it stick out. So they call those helper cells. So in all of this process, in all of these processes, we understanding how our immune systems work. Then the idea of immunity is the idea where we are looking like how to deal in a situation where you have enough of these memory cells or this type of T cells that will recognize a particular kind of pathogen and deal with it. Now, if you already have more of a particular kind of B cell that has receptors that target a particular pathogen, you are better off. Just the same way that when we were immunized, like with the COVID vaccine, it made us have B cells that could recognize the red structures, the red spikes that they said the COVID virus had. They called the spike proteins that were on the COVID virus. So we had B cells that were made to recognize those spike proteins on the COVID virus. If you took your shot of COVID, it exposed you to the proteins, those red spike proteins, and then you had B cells, memory cells that were already, many of them, capable of identifying it, such that when you actually get infected with COVID, you had many of those cells that could recognize its presence, such that by the time the COVID wants to start dividing, those have gone now the clonal expansion of many more B cells to produce antibodies that will go and tag the virus and destroy it. In this case where you have cells, B cells producing antibodies that will go and tag the pathogen for destruction, we said it is active in the sense that it is a process that is going on. Sometimes individuals get so sick that they don't even have enough B cells. They cannot go through the process of waiting for the B cells to go identify the pathogen and then make the expansion. Sometimes they will just take like antibodies that have been made in the lab and give them to those individuals. What does that do? Having the antibodies already gives their person's immune system a leg up, because now they have the antibodies will go and tag all of those pathogens. And when they tag them, it makes it possible for the other destructive cells now to go and destroy them. That, oh, this is tagged, this structured complex is tagged for destruction. So you hear about active and passive immunity. So we said active immunity, an individual body makes antibodies against a particular antigen. It can happen through natural infection or through immunization, which is the part I was talking about, you being given a COVID vaccine. There are certain conditions where you get infected by a particular pathogen and your immune system is strong enough, you got well from it, but those memory cells stay behind. So that is when it happens in the natural way. But immunization is when they give you a vaccine that contains a certain antigen from that pathogen, exposing you to it. I said in this active immunity, the primary exposure is shorter lived and slower to respond, while the secondary exposure is much more rapid and strong response. So when somebody is infected by a pathogen the first time, it's longer, it's slower. Why? Because your immune system, it's almost like they had equal amount of B cells with different receptor types present at the beginning. So when a pathogen is introduced, they say, okay, which one matches? Which one matches? They're still going to find out which one matches. In that way, that process of finding the ones that is the right fit, then to go the clonal expansion, it takes a longer time. But imagine that if somebody who had been immunized had memory cells, meaning they disproportionately had more of a particular kind of B cell with the receptors that are targeting that particular pathogen. When that pathogen comes around, it doesn't take a longer time for the right one to meet and encounter the pathogen. Why? Because there were already more memory cells, memory B cells with that recognition type present. So that one is faster. The situation is faster. They recognize it faster and goes. So let's look at what happens here in exposure in the primary response. Let's say we take even immunization, for example. You have here, when you have your first vaccine against something, let's take the COVID one that we talked about. When we were being vaccinated with the COVID vaccine, to be more specific, they gave us a piece of messenger RNA in that vaccine. We're calling it RNA vaccine. As you're going to see, the messenger RNA, how did they get that to begin with? Those proteins, those red structures that they found on the COVID virus, they took those proteins on them, those spike proteins. They took them, proteins are made up of amino acids. They took them and then they found out the amino acid that made up those proteins. And then they went behind and found out what were the messenger RNA. I think we talked about RNA before because we said it has information that would go and tell which order in which the amino acids are connected together. So from protein, they found the amino acid sequence. From the amino acid sequence that made the protein, they found out the nucleic acid code that made those messenger RNA, that made up the amino acids. So the messenger RNA has instructions to assemble the amino acids together to make the proteins. So what they gave us in the vaccine were instructions to make the protein, that spike protein. So when we took that vaccine, our cells build up that protein, the spike protein. Then because it was strange, it was new, it's something that our cells do not see, our B cells recognize that protein, the one that had the right match B cell, it recognized it, and then it made more of the B cells. Plasma cells that were making proteins and memory cells. But when they found out that interaction, that those proteins were not something that was bad, they destroyed them. And then the memory cells stayed behind. So let's look at when we were vaccinated right here, say at a time day 0, we had the vaccine. And then all this stuff of identifying which is the right B cell to do clonal expansion, it took a while. A few days, you started having, when you had the COVID vaccine, you started having this inflammatory response in terms of having these chills, almost like you're feeling sick. It was our immune system trying to deal with what it saw as foreign. The B cells trying to do stuff, the T cells trying to recognize and deal with it. But all of this, when it happened, the clonal expansion of both the B and the T lymphocytes, it took about 15 days or so for everything to peak out, and then they went and destroyed it. But after they destroyed that fake, the antigen that was presented to us, they destroyed it, but memory cells stayed. They're showing here, this is what the concentration of plasma antibody, because when the B cells were being made, the ones that had clonal expansions, produce more of the antibodies. After that, they had destroyed everything. The antibody levels that were recognizing the COVID went down, but memory cells stayed behind. So let's say after 60 days, somebody, the real virus wanted to infect us. But at this day 60, the person that was being vaccinated has a lot of memory B cells that can recognize the virus. So when the virus infects the individual, those many B cells would easily recognize it. And then because of their numbers, they will start going clonal expansion, and then many of them start making antibodies very quickly. So you see here between this time and within, say, the next 15 days or so, the concentration, this is the amount of antibodies in our plasma, which is the liquid portion of blood. The concentration of antibodies rises very quickly. And that is why the person that had received the vaccine, it's not that they don't get infected, they will get infected, but their immune system will respond so fast and so overwhelming that their symptoms will not be much, and then all of a sudden they start destroying the virus, and then the concentration would go down after the virus is being destroyed. This is actually the benefit of vaccination. The purpose of vaccination is to expose you to the antigens of a particular pathogen, so that you can create more of the memory B and T cells, enabling you such that in the future, should you have an encounter with that pathogen, as the pathogen is struggling to divide, to overwhelm your system, because you have memory cells present already, they would respond even faster and overwhelm the pathogen. When we get infected with pathogens, it's a fight. The pathogen trying to divide as fast as we can, our immune system trying to respond as fast as possible. Whoever wins, if the pathogen wins in terms of numbers and overwhelms our immune system, we are in trouble. But the desired response is our immune system to respond so fast, producing more antibodies, more T cells that have been activated, that would overwhelm it, and then we get well. So we have try to understand this difference of the benefit of having a vaccine, and how the situation of somebody who is comparing somebody who has been vaccinated to somebody who has not been vaccinated, to see how they would respond. We have here, because of the efficiency, the effectiveness of vaccines, we are developing vaccines now against many things, and at different age times from childbirth, we get vaccinated against many different kinds of pathogens, because we have vaccines for them. But talking about vaccines, there have been situations where people are had questions about vaccines, or have doubts about whether vaccines are good or not. I don't know whether this might be a time to which we can clarify to find out whether anyone has any questions regarding vaccines or concerns about vaccines. Any question? How many people here have taken all the vaccines they are supposed to take? Has everybody taken? Okay. Have you taken all your vaccines? You're raising your hands. Have you taken all your vaccines, or you had a question? Just to be clear.
Yeah, I was just trying to say that I had taken my vaccines.
Fantastic. Who has questions about taking vaccines? Anybody?
I do.
Yeah.
Yeah, I took my vaccine. And I'm not opposed to it or anything. But what do you have to say about like reports of like those very horrendous side effects? Um, I don't want to get into like conspiracy theories, but stuff, accidents do happen. What do you have to say about that?
Okay, that's a very good question. I'm glad you brought that up because in the when they make this when they make therapies, we as over if all of us were from one clone, means that we were exactly the same, then none of us will be allergic to peanuts, for example, or none of us will be allergic to wheat or different things. But as a population, we are slightly different. We are slightly different, meaning that even with the food that we eat, some of us will eat it without any issues, but some people will eat it and have issues because of that variance. The same thing goes with medication. Some people might be allergic to Tylenol just because they are slightly different. And if you take vaccines like something that again we take, we even because of the differences that we have, some people would respond to it, might have allergic reactions to them, and in that case, it will not be the suitable thing for them. So to answer your question generally, I'm saying is that the safety of something is usually determined about not whether 100% of people who take it never have issues. It's more a function of what percentage of people would take it and not have issues. Because the thing, when they take a vaccine, when they are making it to test whether it is safe, is they would take a population, like a sample of many people in the population, race, gender, age, and even other factors. And then they take a sample of these individuals and give them the stuff. And then if it works 95 or 99% of the people to 95% of the people, they would say, okay, well, if they know the attributes of the people who had problems during that testing period, they would say, well, this medication is, or this product is available, but if you have this and if you have this, we suggest that you don't take it. So, but sometimes the people that they get exposed to, because it's not, if they exposed everybody during the trial period and none of them had issues, and then they turn around and say, okay, this thing is safe to take, but they took only a sample of the population when they were doing those optimization or those during the testing period. So you could see a situation where somebody, even though they had said something was harmless to a bunch of people or to most of the people who took it, and then somebody takes it and then they have issues. So the dilemma we have is nobody ever says that something is 100% effective. Usually the situation is that it is safe to take and it helps a majority of the people. The data is out there and it's almost like it's your choice to take it or not take it. So but the question to also ask is, if you have something that could help 95% of people, and then 5% of people or 2% or 1% of the population gets sick from that thing, how would you deal with the situation? We had COVID before. Would you say that, oh, well, because this thing 1% of the people get sick with it, then we should not give it to people. That is really the challenge. How do you answer that, Vladimir? What do you think they should do in that case?
You mean if 95% of the people could help the other 5%?
No, that means if something is effective for, say, 98% of people, but it has the chance that in some 2%, some might take it and then they have problems with the thing. What do you say?
Sorry, I'm on the hotspot. I don't know. I got to think about that. Sorry.
So the thing about what the way I would say is that the problem becomes when, for example, the concept of vaccine was very, because they tell you in advance and say, it works in this percentage of the population that they tested, it worked out. And then they tell the health department, well, now we have made this thing, it works in 98% of the people, use it as you see fit. But the bigger problem was that some people also do is that if they, most of the, but so this concept of being transparent in terms of the efficiency of a particular therapeutic, whether it's a vaccine or just even regular medication, people had bought into it. That is why we take Tylenol. That's why we take medications. That's why we took vaccines. But the bigger issue comes when somebody says that, for example, I took a vaccine and then I had cough. But if you know the way the vaccine works, you know that even if you're going to have some kind of symptoms, cough might not be one of them. And then somebody says that, well, I took a vaccine on this day and I had cough. So people should not take this vaccine because it causes cough. To be more direct, there was a reputable scientist from the UK had associated kids being given vaccines to development of autism. Somebody who is autistic had certain they have certain features, and they tied it. The guy tied it that it was because those kids had been vaccinated that they had autism, and people thought for a while that was true. But unfortunately, they could not replicate that guy's finding in his studies. So when the word went out, people who unfortunately had kids that had autism blamed it on the vaccine. And how did this even come about? Was because when we talk about also consuming scientific information, is that because the guy was somebody who was a reputable scientist and had done more studies and reported them with legitimate experimental procedures and observations and conclusions. When he did this study this time and reported it, because the process of you having your scientific discovery reported is that when you do your experiments, you write your data, and then you send your experiments, the results, and the conclusions to a set of experts in that field to review it. When they review it, then they publish the data and the experiment and the study in reputable journals. But just imagine that somebody I have been doing this and published like, say, 50 papers of 50 different studies, and they were very good. And then somebody in another somebody had only done 2 papers. When the person is sending in their third study experiment for them to publish, and I send mine that I've done up to 50, the team of experts that are reviewing it will not scrutinize my paper because they think that, well, this is an experienced scientist and everything 99% of the things that he provides us is usually hot, his processes and his techniques of experimentation are very good. So they don't spend too much time analyzing, scrutinizing my own stuff as they would somebody who is just doing it, doing their studies for the first time or second time that they're trying to publish stuff. So in this case, about this autism situation, a reputable scientist that had contributed a lot in other things made this study, but because there was a quest, there was a big desire to trying to understand what causes autism. So he just came up with fake data and then gave conclusions or results and conclusions to tie vaccines to autism. Because when people started trying to replicate, because when you do report your experiment and you say the way you do it, what you require, the process, the procedure, when I do it and you do it or somebody else does the same thing using those steps and those methods, we should all come out with the same results. But since that guy in the UK did his study on autism, nobody was able to replicate it. Then they started pointing out, some people say, well, wait a minute. If none of us are able to replicate these things, let's go a deep dive into how this guy did this study, his results, and whether the results would match the conclusion that he came up with. And since then, they found out that he fabricated data and that it was not a good experiment that he did. And then they kind of downgraded his reputation from the scientific community. They pulled down the paper, the study, but by the time they did that, the explosion of his results regarding the relationship between autism and vaccines had gone out viral, and many people just thought, even they'd come back to tell them that, oh, this guy's data was fabricated, his study was bad, nobody's buying it anymore. So he created a situation that casted a lot of doubt about people taking vaccines and their benefit. So that is one of the challenges of, and that through classes like this, that you're able to scrutinize stuff when they say this thing causes this or doesn't cause this. You're able to ask more questions and then see, to be able to make better decisions. But to answer your question correctly, the dilemma is when because we are slightly different of the variations that we have in our population, it is inherent that some things would affect individuals in a different way. But the goal is to always make that anything that we are giving like a solution to a problem is overwhelmingly of benefit to the population than if it's going to cause harm. And I think that is the process that they've been used, whether it is medications, whether it is vaccines. Any question? Okay, I'm just going to go now very quickly. Let's finish this part here. Immunity. It talks about, I say in passive immunity, an individual is given prepared antibodies against a particular antigen. Passive immunity, the mother gives milk that has antibodies to the baby because the baby's immune system is not producing it. But the deal is, it is short-lived in the fact that antibodies just go there and attack the pathogen and it is destroyed. The antibodies don't stay forever. I said this can happen naturally as antibodies are passed from mother to fetus or artificially via an injection of antibodies. When some people have cancer, sometimes they would have they take the cancer cell and they will look at the specific antigens on it that are unique to that cell. They will make antibodies in the lab, and then they will inject that person with the antibodies so that they will go and attack those cancer cells for destruction. Our ability to make antibodies in the lab is part of the application of understanding how the process of the immune system works, and then also cancer cells. So if you think about a cancer cell basically is a cell that has the characteristics of dividing in an uncontrolled manner, meaning that the cell will divide and then you keep dividing, keeps dividing. Ordinary cells would know when to stop, but a cancer cell does not. A B plasma cell, on the other hand, is a B cell that could produce antibodies. So imagine, as we saw, this is an example of a B cell, keeps producing these kinds of antibodies. This is an example of a cancer cell. Somebody found out that you could take this cell and this one and fuse them together, which is what they did. And then this cell that is producing, B cell that is producing antibodies, this cancer cell that is growing together, when they fuse them, they form a hybrid cell, which is called a hybridoma. This hybridoma now has two capabilities, those of both cell types, capable of growing continuously and shedding antibodies. And the shedding antibody part comes from the plasma cell. The growing unstoppable, that means you keep replicating it, comes from the cancer cell. So in this way, when they grow this in the lab, they could just purify these antibodies and then concentrate them. Then they can inject them into somebody who is sick. So if somebody had a cancer cell, as I was indicating, a cancer tumor, they take some cells from it, they look at the B cells that are producing antibodies for those receptors on that cell, they go and grow them in the lab, fuse them with a cancer cell, and then produce many, many, many antibodies from it. And then they are able to take these antibodies and inject them into the person. These antibodies will go around in their body, and each time they see a cell that has those cancer cell features, they would bind, they would tag them, and then the destructive cells will now come and destroy that particular cell. You can watch these links and then see how the process works. How can the immune system react, and that in a way that can be harmful to us? Allergies, tissue rejection, and immune disorders. For allergies, things that are normally not would not cause our system or cause any harm to us, but for some reason, our immune system cells, they think that, oh, this is something that is going to cause problems. And then the immune response comes to play. The inflammatory response happens. So just imagine what we were talking about. Histamines being produced, blood vessels dilating, and then bringing in more white blood cells to move into the tissue. When somebody has allergy responses, those are the features that those are the symptoms that we see: inflammation, redness. And then if it is somebody that has asthma, for example, they would have that things can cause restriction, inflammation to cause restrictions on their airway. It causes problems. So if your body feels like it is being threatened by something, and it turns out that that something is something which should not harm you, then it could happen that way. And if that response of vasodilation is systemic, that means it happens all over your body. In a wound area, it would happen and your body would swell. But if all your blood vessels just got bigger, your blood pressure would drop so low that you can die as a result of it. So allergy reactions are example when we have our immune systems going awry, and then the other one is tissue rejection. So you can see cytotoxic cells, for some weird reason, the T cells that are supposed to have learned the characteristics that happen on our tissues, or that are the things that are surface proteins and carbohydrates that are on our own cells and our tissues. For some reason, they lose that knowledge and they start attacking our own cells as if they were something that is the foreign. And then some of these things they have to take immune. For people who have allergies, when they run into these situations, they have like antihistamines that they take to counter the effect. And then these ones, they have tissue immune suppression drugs that they to help them in that situations. But sometimes tissue rejection also happens in an inherent situation where somebody might have a damaged organ or damaged tissue, and they require tissue from some other person to be able to keep them alive, so they will do transplanting. So you are better off taking the tissue or the organ from somebody else, but now they would always have immunosuppressive drugs to make sure that their immune system does not totally fight and destroy that tissue. And then we have some autoimmune disorders like multiple sclerosis, lupus, myasthenia, Graves' disease, and arthritis. All of these are situations where our T cells are attacking our own body cells. And then we have immunodeficiency disease. It means our immune system is not strong enough. You remember we said, like, in certain situations, certain pathogens need to, T helper cells would go and engulf the pathogen and then present a piece of the antigen on its surface for the destructive cells to identify it and destroy it. In AIDS, for example, the cells that are destroyed, the virus affects but the helper cells. So when the helper cells cannot recognize the pathogen just kind of goes around, keeps dividing, and then the person has issues. So below here is a summary of again, the adaptive response, which is the one that is specific. And the second slide here it just takes about the general immune system. It talks about the external defenses that we have. And then it talks about the internal second line of defense, all of these being non-specific. And then the other one that is called adaptive or acquired immune defense is the third line of defense, which is characterized by the fact that it is specific on the pathogen or the antigen it is dealing with, and it involves B and T cells. Question? The one thing that I would like you to, how I want you to, or that I found that people remember this easily is, we have many names of many things here, of different cell types coming into play. If you first understand the process, like in the first video that I had showed you, what I said was introduced to like fifth graders, you kind of just understand the big picture of how things are. In terms of, you have our bodies, things that prevent things from getting in, barriers, chemicals, where they are, what kind of chemicals, what kind of barriers. And when you are preventing something from getting, it's almost general. You don't, any kind of thing you are trying to prevent it from coming in. We have things that do that. First line of defense. Second line of defense. Some stuff gets in. our body just goes and has cells that goes and deals with the new stuff that just comes in. It's not specific in terms of what it's exactly dealing with. It's just saying that this stuff doesn't belong to the body. And we have the macrophages, the neutrophils, that inflammatory response. That is the second line of defense. It did not prevent stuff from getting in. Stuff has already gotten in. But sometimes whatever has gotten in, that second line of defense is still cannot solve it. Then the third line of defense, which is a little bit slower, but it can go now and identify and hone in on that particular thing. And once it hones in on it, it makes antibodies. It has T cells that go and destroy the stuff. And then they have memory of that particular encounter, of that particular pathogen. And then when you have that big picture, you watch these videos, then the rest of it, the names of the things will come to play more in terms of being able to understand it. So this chapter took the whole time we're supposed to meet today. And I deliberately made it so that we don't rush it through. But if you have any questions about this, let me know in any time between now and when we take the exam. We'll have this chapter. I will have a quiz for you on this week. The quiz will be on Thursday, on Friday, so that it allows you time to be able to start reviewing this quiz on this one. So, any other question on this before we move on to something different? Some people misunderstood that we are taking the exam today. Yes, you'll be exposed to the exam today, but you don't have to. You are not taking the exam in the lab today, no. So when we get to the lab, if you have any questions regarding the lab, regarding the exam, you would ask me. If not, then I will allow you to be able to start doing the exam, not in the lab, but you will do it. You will have it available for you to do tomorrow, and then through Tuesday, you will do it at home. Last week, we talked about the digestive system. I will let us, the other system that I would like to talk about after as we complete this, as we've completed this immune system, lymphatic system, I would like to also talk about the respiratory system. And I'm saying we should talk about it because it's after we have I said that you would need to know how the system, we cannot separate the system is to like the upper and the respiratory system, because the rest we have our area, our nose and our pharynx through which we breathe in and air, and then inside lower we have our lungs. So if you think about how air moves into us, we have our nostrils through which air passes. It comes through this pharynx area here, because that's the area where it connects from the mouth and from the nose. We have this area right here. And then we have the trachea right here. This rings of cartilage that keeps it open. It comes down as a tube here that branches here into a smaller tube with smaller branchlets here. So you have the trachea, the bronchus for one, bronchi for two of them. This branches into two lungs, which we have, and then fans out into bronchioles, smaller branches that end up in sac-like structures called alveoli. And these lungs are within a thoracic cavity. Underneath it, there's a sheet of muscle called the diaphragm, and they reside in the pleural cavity. So the passage of air is from the nose through the pharynx, the larynx, and then the trachea, the bronchus, the bronchus, and the alveoli. So in our nostrils, we have a lot of blood vessels that are red in the surface. That means if you pick your nose, if you have like stuff in your nose and it gets dry and you pick it sometime, it can actually make you to start bleeding because they have capillaries right in the surface of the surrounding of our nose, and the capillaries there are bringing blood there to be able to make the air we are breathing warm. Because if you are freezing, the area is cold, we don't want to suck up cold air into our lungs. So one of the functions of the capillaries that are in our lungs here is to warm the air as we breathe in air. We have hair-like structures inside our nostrils and mucus, the hair to trap particulate matter, the mucus to trap stuff so that particles don't get into our lungs. In our pharynx area here, it allows that you can also open your mouth and breathe through your mouth. And then one cool thing that we have here is an epiglottis. This epiglottis right here, this thing that would cover, and if you are eating stuff, it will take this flap here, would cover here, such that food does not come into your windpipe. Back here is your digestive tract or esophagus. So if you are eating and talking, this stuff doesn't work very well at that point. So that is why they say it's not usually a very good thing to eat and talk, because if you are talking, this epiglottis might mess up and not close properly, and food could get into your trachea here, which is going to be a very bad thing. Now we said here the trachea here, it branches like this. The right lung tends to have like 3 different lobes, 1, 2, 3. Meanwhile, the left lung has 2 lobes. A better structure here. We had mentioned tonsils here because, again, stuff that comes in through our nasal passageway. If there's some infections that are trapped in here and it starts getting inside our tissue, these tonsils that have white blood cells would be alerted to try to send white blood cells to deal with it. The tonsils provide lymphatic defense during breathing at the junction of the oral cavity, and all these tonsils, they're able to do this. Why? Because they have white blood cells in there. They talk about the trachea here, I talk to you about the epiglottis here. And then I said the olfactory, sometimes we have when you cry, your nose runs because we have connections here through this passage that comes into our nasal cavity and things like that. Tear glands. This is what I just indicated here. This is another way of looking at it from here. We have our voice box here. Again, we have this triangular cartilaginous where air passes through between the trachea and the pharynx, and it vibrates to make noise. That's how we're able to speak. But it's an opening, which again, when we are trying to talk and we are trying to eat, Then this glottis that is epiglottis that is supposed to close it, it doesn't manage very well, and food can get through in the wrong passageway. Now, if you look at the architecture or the structure of the trachea, it's almost like those carpets that used to have like threads sticking out. So in the inside of the trachea, you have cilia. Embedded in them are goblet cells that produce mucus. The cartilage we know that cartilage is slightly flexible, it's connective tissue. It maintains the structure, it keeps the structure of the trachea open. The goblet cells produce mucus. Mucus and the cilia together trap any particulate matter, and so that when material is trapped here, you start having irritation where you gag and then you feel like coughing. It causes this reflex where when you cough, sometimes the stuff will come out, mucus will come here, and then when it rises up here, if you are in a place where people are, you just kind of swallow it, because when it comes off here, then you could swallow it. It closes here, and then you swallow into your esophagus. Or if people are not there, then you could cough it out and then throw it out. At the end of the bronchioles are sac-like structures called alveoli. One is called alveolus. I mentioned the number of lobes that the right lung has 2, the left one has 3. This is right, this is left. And I said the membrane, so like where our lungs expand and contract, and the pleural cavity, the space in which they reside, they have a pleural membrane that kind of lines that cavity, makes the lungs to slide with less friction as they go up and down, contracting and relaxing. I said within the trachea, within the bronchioles, they end in sac-like structures called the alveoli. This is like a bronchiole. And then this round is almost like a branch of grapes. Just imagine the grapes with these coverings, vessels covering them. And these capillaries are 1 cell thick, and they cover the alveoli. Being 1 cell thick and the alveoli very thin, it allows for exchange through diffusion. Material from the blood will diffuse. The material, in this case, carbon dioxide, will diffuse from the blood into the alveoli space. And then, since the blood that was coming into the air that was coming into the lungs was from the outside, it proportionally has more oxygen than the one in the blood. So oxygen would move from these alveoli spaces into the blood vessel. Carbon dioxide does the reverse. More carbon dioxide in the blood that is being brought to the lungs through the pulmonary artery. The carbon dioxide would diffuse into the alveoli space, and it is taken back out as we breathe out. So in the process of breathing, we have inspiration, where we suck up air, where we take up air, and expiration, where we breathe out. What is involved in inspiration? When you if you breathe in, certain things happen. This is our rib cage. In between our ribs, we have some muscles they call intercostal muscles. When they contract, when they contract, like when we are breathing in, our chest rises and expands because our rib cage would rise and expand. And while it is doing this, this other lower sheet of muscle called the diaphragm also contracts. Normally it sits in a bell curve shape, but when it contracts, it flattens instead. And when it flattens, the space, and this one is expanding, the ribs are expanding, the lungs turn to expand, the pressure in the lungs is lower, then air rushes in. That's why when you are breathing normally, just breathing normally, that is what is happening. Your chest keeps rising and lowering, lowering, rising and lowering. So when you are during inspiration, when your rib cage increases, contracts and rises and expands, the pressure inside here is lower. Air rushes in. When it contracts, when things come back to normal, the space here is reduced. It's almost like presses onto the lungs, and then the air inside is forced out. Expiration, again, is the reverse of this. So in talking about some of this breathing when there are some things that we have to consider. Some things you ask, okay, is our lungs always, is there air in our lungs sometimes, or when we are breathing, but can we suck extra air? Sometimes people who are trying to understand how our lungs are functioning, it becomes important to understand this. So I said they will talk things about the tidal volume, which is a small amount of air that usually moves in and out each time we breathe. Because normally we have air always in our lungs, or else our lungs will collapse. So with the existing air that is in our lungs, called residual volume of air, the extra air that comes in when we are breathing and goes out is called the tidal volume. So when we talk about the capacity, we say the maximum volume of air that can be moved in plus the maximum volume of air that can be moved out. So imagine that normally we are just breathing normally, and then I say breathe in some more. And then they say breathe in extra, you breathe in more, and then say breathe out. And then so that maximum amount of air that you can breathe in and out is called the vital capacity, and it helps people who are looking at how well your lungs are doing to understand how when they do all of this exercise using a spirometer, they're able to tell how well your lungs are functioning. The inspiratory and the expiratory reserve air is the increased volume of air moving in and out. So I said when they're trying to visualize this, you can look at this is normal breathing, and then the extra inspiration and extra expiration is they kind of measure this. They use this spirometer to be able to do this. They're able to understand how well our lungs are functioning. Now it begs the question, what controls how we breathe? We breathe autonomously, meaning that our brain talks to our lungs they sense the amount of oxygen in our blood, they make us to breathe. So I say nervous control. Say there's a center in the brain called the medulla oblongata that would send out nerve impulses to muscles to contract. So this area here goes, I talked about the intercostal muscles and I talked about the diaphragm. They would send communication to these muscles. The medulla oblongata will send information here, contract and relax. We do this, we are not aware of it. We know we are breathing, but you don't say, I'm breathing in, out. No, it just happens. But in having this autonomous control of how we breathe also has some issues. Like we have some situations where babies die in the crib while they were sleeping, and the understanding or the explanation they suggest is because this center that automatically controls how they breathe, something happens to it and they don't breathe, and they are unable to breathe. And then I said there are also chemical controls, because when we have situations where there is high amount of carbon dioxide in the blood, carbon dioxide will combine with plasma or water in the plasma to form carbonic acid. Acids will cause the pH in the blood to reduce, and systems, receptors that would sense this would now enable the place in the brain to send more impulses for us to breathe. I said 2 sets of chemoreceptors sense the drop in pH, one in the brain and the other one in our circulatory system. I said both are sensitive to carbon dioxide levels that alter the pH in the blood, and will now inform our breathing muscles to engage in breathing more as needed, to be able to bring in more air. When bringing in more air would move out more carbon dioxide and enrich the blood more with oxygen to bring up the pH from where it was low, getting low. And then when you talk about gas exchange, you'll hear something about partial pressure of carbon dioxide, partial pressure of oxygen. They're just comparing, for example, if you are comparing the amount of oxygen and carbon dioxide in the air that we are breathing in, and the amount of oxygen and carbon dioxide in the blood that is being brought from our bodies from the heart to the lungs, then you could understand in which direction the oxygen will be moving to and which direction the carbon dioxide will be moving to. Normally, the carbon dioxide will be moving out of your blood vessels into the alveoli space, and then the oxygen will be moving from the alveoli space into your blood vessel, your capillaries. So I said the exchange of gases between the lungs and the alveoli and the blood capillaries. That's just what I was explaining here. How carbon dioxide is carried. Carbon dioxide combines with the hydrogen and the bicarbonate, forms carbonic acid, then it breaks down carbonic anhydrase, breaks it down to release carbon dioxide into the lungs. And then oxygen, again, binds to hemoglobin in the blood. This is an important picture to remember when we talk about all this partial pressure and things. Imagine that this is our heart right here. The blue ones is blood that came from our body, and now the blood is brought from our body to the lungs, and it is blue because it is rich in carbon dioxide. And the heart has to send the blood through the pulmonary artery to the lungs, where it's supposed to get rid of the carbon dioxide. And getting rid of the carbon dioxide, this is what happens here. So you're in your lungs, you have the blood vessels, and then what happens in the blood vessels? Carbon dioxide is moving out of the blood into the lungs, and oxygen is coming in. Oxygen gets in, and oxygen goes into what binds to the hemoglobin and is brought back to the heart in the pulmonary vein, and then it goes out to systemic circulation. So remember this diagram. Again, most of the ways in which oxygen is carried. Some diseases that would, infections or conditions that affect the respiratory tract. If you're talking about our nose, our mouth, and our things, sometimes we have sinusitis, where our sinuses are blocked. Either they're inflamed because of infections, or sometimes we have conditions where our inner ears are infected, or our tonsils gets inflamed, or our larynx gets infected as well. So all of these are conditions that could affect part of what we call it the upper respiratory system. Meanwhile, the lower respiratory system, where things that will affect like our lungs, you have pneumonia, where you have thick fluid being built up in our lungs. You have tuberculosis, where you have a bacterial infection that infects and creates these structures that call tubercles. And then you have fibrosis, where you have, instead of you have fibrous elastic tissue building up in the lungs. And then emphysema, where your alveoli are damaged. Asthma, where your bronchioles become narrow or irritated, and narrow because they are inflamed. And then you have wheezing. And cancer, when you have the tissue there, starts growing in an uncontrollable manner. This is a better diagram that illustrates that. Pneumonia, you have fluid build up in here. Fibrosis, you have connective tissue being built up in here. Tuberculosis, you have tuberculosis, where you have these materials, tubercles, forming in your alveoli. Emphysema, where these alveoli, they break, the walls break, and then you have like a bigger hole. And the reason we have these small bunches of alveoli is to increase surface area for your blood vessels to do the exchange of gases. And then emphysema, you would see here, this is the bronchi. When they get inflamed in asthma, you have these passages become narrower. And then when you are breathing in, they start creating but sound as you breathe in and out. Bronchitis, you have again inflamed situations, either an irritation and the buildup of mucus. Some of the things that, in terms of health focus, that could affect our respiratory health is when we smoke stuff, when we inhale stuff, we are supposed to be breathing clean air without particulate matter. Unfortunately, sometimes when we smoke cigarettes or smoke other things, we breathe in materials in whatever we are smoking or inhaling, even from the environment. So smoking increases a person's chances of lung, mouth, and larynx, or esophageal, all of these cancer types. These other parts here is because when you introduce some of the substances in the smoke and they get into our blood systems, they could move and go to this other, whether the kidneys, the pancreas, or the stomach, or even cervical cancer. When people have lung cancer, I said the survival rate is only like 13%. When you smoke, it increases chances of emphysema, bronchitis, and all of this. The worst part, if you are pregnant and you're smoking, it can affect your developing baby. And smoking is one of the things that there have been no advantage to it. Now there's a lot of awareness now that they don't smoke. If you go on campus, if you're in Dianza, they create some kind of a bench somewhere in some part of the campus for you to go sit there and smoke, because in the past, people used to smoke inside buildings, smoke in airplanes, but because of the awareness of the harm smoke can cause to us, people are staying away from smoking. I kind of always put this, that when we talk about the impact of some of these things, and then the response to ban them, and you weigh them against the rights we have to even do harm to our own bodies, there's always this friction between what right do you have to ban me from smoking if I'm smoking in the basement of my house and I don't bother anybody. So I said, does restricting the freedom to smoke segregate people based on habit? Okay, can we also ban people from drinking too much soda? If you ban me from smoking? If you say that I smoke and I will cause, because I share insurance with you, I will cause your insurance premiums to increase. Am I going to also say we should restrict you from drinking soda because we know that soda can cause is bad for you? It can cause your blood sugar level to go high. So we have some of these things, decisions which we make, and our policymakers that make legislation or pass laws, we influence them to be able to make some of these decisions. But some of these habits that we have individual and societal consequences. And sometimes some of the policies that we make through our laws, the laws that we make, sometimes we always have to balance them towards the rights that individuals have against, and then trying to make sure that we are also not discriminatory in terms of habits that we don't do and try to ban them. And then while we have habits that are not necessarily good for us and still affect, if we're using insurance as a way to say that because it's affecting our cost of insurance, we should cause these restrictions. But we know now many aspects of some of these habits that we do that affect our health. So I said, do smoking bans hurt the economy? So we have all of these things, and it's something that we can always have a discussion about. But this is just a good summary of how our respiratory systems work, and with a goal of trying to introduce oxygen that we need for cellular respiration and get rid of carbon dioxide, which we make as a result of cellular respiration. Does anyone have any question?